This video is really nice.
So we can start taking up questions from the chat box.
Yeah, now we can start questions. Before questions, presenters can little bit, if they want to say something, they can say for like 2-3 minutes. Bivas, Kunal, Mandar, Batul, want to say anything on the presentation?
Yeah, so like after the lockdown, like we were working in the Cube Lab maintaining cultures of CLA, soil nematode. But after the lockdown, I think both Anshu, Batul ma'am, and along with the other collaboration of the Cube, they have found a new substitute for culturing the same thing in our home with the help of potato as a medium. And I was also going through the questions. So yeah, as Nagarjuna said, rightly mentioned potato as a new petri dish. Yeah, so it's a collaboration effort from all over the places, people joining us all over the places. And also the video is itself made by several people. Like the animation is made by some people, the memes are made by some people, and also how the sequence is going to be. So everybody participated and worked in a group wise manner. So yeah.
YouTube stream has started.
So I think we can start taking up the questions. So you highlighted the point. That there is no such discovery of nematodes.
Streaming is a start.
Hello.
Yeah.
Yes, Jay.
Anybody had question? Some question?
So one of the question is that in the video it was portrayed that in India we don't find nematodes, especially C. elegans. So how far is that true? Like for
Okay.
some years,
So answering to the question, the video said that from India it is still not discovered. Like even in the poem in the beginning, it says that. And how true is the thing? I'm going to say, like, there are several papers. I'm going to quote the papers as well. Paper work done by Marianne Felix, where one of the researcher who is working on C. elegans, and she has published a paper in the Cell Journal about the worldwide distribution of the C. elegans, in which she has surveyed the soil from the coastal region of India as well as the regions of the Gujarat. And in that reference paper, how are we sure when was that paper? That paper? How are we sure? Okay, so yes, the thing is, like, no paper quotes that there is the presence of C. elegans in India, but only one paper which says that they haven't found. So we are not very sure that whether C. elegans are there in India or not there in India, but just it is an effort to like if we can find out C. elegans. And the paper was on 2007 paper published on the Cell Journal.
So so one of the question is, you mentioned there are several Nobel Prizes going to nematodes. So what is your question? What do you want to study on nematodes? Hello, was I audible?
Yeah.
Yes, Saida.
Yes, so our long-term objective is to find out C. elegans from the Indian native soil, and once we do that, like it's a long-term objective, and currently we are immediate objective, if you ask, is like how to maintain and sustain the culture in the home lab, where Anshu and Bartlebam has tried several methods, and we are trying to tinker about several small steps that we can find out and maintain and sustain the culture in the potato as a nutrient, as an agar medium for culturing nematodes.
So why agar on the first place? Like, why do you need agar? It's found in soil. So why not just directly take it from soil?
So, talking about the agar, when we are in the lab, we use agar to culture nematodes, or to basically culture the nematodes. So, what we do is, it has 98% of water and only 2% of agar into it. So agar is to like a solidifying agent which will give a base to it to crawl, and as it is found in soil, it also needs water. So the 98% which I said is the water. So it gives its moisture, and so it can like crawl and live on the plain agar media. So that's the reason for using the agar.
And also another reason for using agar, not soil, is like if we keep the nematode in the soil, culture the nematode in the soil, we will not be able to get a single line culture that is very essential for maintaining the nematode. And we considering there can be other eggs as well as other strains of nematodes present in the same soil. So one of the reason maintaining in a plain agar culture is also moisture is needed. So this two combines to maintain the culture in the agar.
And also I like to add on that we can observe the nematodes more clearly into the plain agar, and in soil it is very difficult to observe the nematodes through the microscope. So it gives a base which is a transparent base, and so that passing the light we can use a microscope, whether it be a compound microscope or a dissecting microscope, we can observe the nematodes.
So Kunal, I have a slight question. If you can hear me. Hello.
Yes, Shivam, we can hear you.
Yes.
So I just wanted to ask about that. You all are culturing all these nematodes in agar. So what does you give them as food? So what do they eat in agar?
So yes, talking about the feed in soil, as they feed on the bacteria that are present in the lab, we also feed them with the E. coli bacteria, to be specific, E. coli OP50 culture which we had. So we used to feed it to the nematodes.
You had mentioned in your video that one of your GoPups was not maintaining enough moisture in the cultures. So how did you overcome that? How did you maintain moisture?
Sorry, can you repeat the question, please?
Maintaining moisture, one of your goof ups that you displayed on in the video at least, that you are unable to maintain the moisture. So how did you overcome that? Like, what did you use?
Which moisture are you talking about? Like the agar plate one or the potato medium?
I think it was regarding agar.
No, it was about the potato medium. First, in the goof up, we are in the home lab. We are trying with the potatoes, so it was drying within 40 hours or 48 hours, I think, within 2 days. So how we overcome that? I think Manasi is asking about that.
So answering to that, I would say like Anshu would correct me, like she has done the thing. But what we are doing is like we used to maintain the uh, potato slice on a wet tissue paper so that the moisture retains, and that's the only thing we were doing. Anshu, do you want to add something?
No, actually, yes, we were just keeping the moist tissue paper below the potato surface so that it will remain moist.
So why boiled potato? Why not raw potato?
Okay, answering to that, I will start answering, and Anshu can continue. We have started with boiled potato as well as we have started with uh, normal
Raw
potato as
potato.
well,
Yeah.
raw potato as well. But considering raw potato, like it actually got fungus more quickly as compared to the boiled potato, so we shifted to boiled potato. Anshu can give a descriptive answer.
Yes, actually, me and Batul ma'am, we both are doing on this setup. So I used boiled potato, and Batul ma'am has used raw potato. Yes, mostly bacterial and fungus growth we observed was after within 48 hours. Means not so quickly.
I
Which
would like to add
media?
one more
Yeah, yeah, very
thing,
much.
that the raw potato, it has starch content in it, and the starch is a polysaccharide. This means that it has long chains which are connected to one another, and the organism certainly cannot, get the nutrient out of it rapidly. So, in the case of boiling, what happens that all the bonds which joins the carbohydrate molecules together, they break up and makes it easy for the organism to get the food out of it. So that is one of the reasons that why we used boiled potato over raw potato.
So are you feeding them potato? Hello? Hello?
Yeah, Jay, you are audible.
So I'm asking, are you feeding them with potato? Is it a feed?
No, sir.
So
Potato is just a surface.
we are feeding them.
Okay. No, actually, we are using potato as a surface for them to crawl, and as a feed, we are using 1 drop of milk at home because we are not we don't have E. coli culture at home, so we are using one.
So nematode cannot eat boiled potato. Hello?
So which medium used to culture nematode? Is there only agar and water we use in media? Is a question asked on YouTube.
No, my question was, does the nematode not feed on boiled potato? Can you grow nematode directly on boiled potato? Because it also has nutrients, right? Hello, I'm audible?
Yes, yes, Jay, you're audible.
So you
Hello.
got my question?
Hello.
Yeah, it was audible to me. Yes, Anshu, say.
Hello.
Yeah, say.
Yeah, Anshu, you're audible.
So nematode feeds on bacteria. So we can't say that it feeds on potato, as there can be bacterial growth on potato also. So maybe they can feed on those bacteria.
No, why not? Because see, bacteria also has nutrients, right? So they take up the bacteria, they take the nutrients, right? So that way, they can take potato nutrients also.
I just
Have you tried nematode on the directly on the potato? Were they growing?
Like a control setup.
Yeah, like a control setup.
Actually, Jai Kishan sir, first there was a problem to identify, to observe the nematode in the potato. First, there was a very huge problem to identify the nematode in even in the soil one. So we were struggling. They were the Anshu or Batukan was struggling to find a nematode first. And in the first setup, I guess they didn't found nematode. Am I right, Anshu? Can you correct me? So they were just struggling to how to identify nematodes on the potato with the naked eyes. Because in the agar plate there was contrast. The agar was fully transparent, but here on the potato slice there was a little struggle to identify exact what will be the nematode or it will be another organism or not. So there was a struggle. Afterwards, they found some nematode. So Anshu, can you add on?
So what is a nematode? How do you describe a nematode? How do you say this particular organism is a nematode?
So, Saida, I have one point to add. I have tried with raw potato. When I tried with raw potato, I didn't see any nematode. After 6 hours, the potato started to dry and there is no moist content in that. So I didn't observe any type of organism in that.
We have a basic
And
definition.
I
What
think the
is
reason is
nematode?
quite obvious. And I think the reason is quite obvious because in the soil there would obviously be much bacterial growth as compared to the potato. So why would the nematode would leave that food source from the soil and uh, come to the potato?
And also, we didn't try picking up a single nematode and placing it on entirely uh, potato on a potato slice, and also um, to see that whether the nematodes is actually eating the potato or eating the bacteria. We have to maintain the potato in a more sterilized condition where we will prevent the growth of any bacteria, because we can't find out whether the nematode is eating potato or eating bacteria, because if we are keeping it open, there is a possibility that the bacteria can grow on the potato. And if by chance the nematode survives on the potato, we can't say that nematode is feeding potato, because there are possibility that bacteria culture has grown on it. So for that, I think we need to maintain the setup with potato and a nematode on a sterile condition to cut the chances of growing a bacteria, and so that we can entirely focus on whether the nematode can survive solely on potato feed or not.
Or you can also try in the liquid, because liquid medium there you can control easily, right? The liquid you can cover and all, and you can
Yes,
try
yes,
putting
yes, we
some
can do
nematode
that.
in that and check whether it grows.
Yes, yes, we can do that. Definitely, we can do that.
I just wanted to ask, like in labs, did you use aseptic techniques and sterilization, and in home labs, didn't you use it, or it wasn't? What I can say, it wasn't working, or did you try to use pressure cooker or anything like that, or to get a replica what you use in your scientific labs? Please
Oh,
answer.
okay. So can you tell me like aseptic techniques you're talking in which step? Are you talking about like while we are maintaining the nematode in a agar plate, or step we are talking about the aseptic culture?
Yes, I mean to say sterilization of media. Like I don't know what is the media for
Okay,
nematode.
okay.
So
Okay.
sterilization techniques are maintained in scientific labs, and do you maintain that same thing in your home labs?
Okay, so okay, yeah, I will answer that. So in the cube lab, when we used to maintain the culture of nematode, what we used to do is we used to take plain agar plate. So plain agar doesn't have any nutrients, so we don't used to strictly follow any sterilization thing, or rather what we used to do is like we used to boil our agar on a simple like pressure cooker, and because considering it's a plain agar, the chances of getting contaminated is pretty low as compared to the nutrient agar. So we strictly don't use any sterilization technique in the cube lab, as well as if you see in the case of what is the drawback, or what is the drawback of maintaining a culture in the home lab is yes, as I said, like potato rots after 48 hours, so there is always a possibility that like we haven't had really found that how we are going to sterilize the potato and keep that particular thing sustained for a longer period of time. So we haven't used any strict norms or any strict methods of sterilizing things in home as well as in the cube lab as well.
So, first question would be, how do you identify nematode, that this is a nematode or not, a particular organism to be a nematode, and how are you observing it in home labs?
Sana, what we used to do in Cube Lab, that how we isolate the nematode. For isolation, we are using soil. So we directly keep the soil at the periphery of the agar plate. If you have a agar plates and you have to extract nematode, so what we used to do in lab, at the periphery we used to place a soil sample, and at the center we used to give us bacterial suspension or the supernatant of that same sample. That the at the center of the plate there will be food for the nematode, that is bacteria, and within 24 hours the nematode used to come at the center and feed on that bacteria. So what was happening? Actually, we were not following the sterilization technique, because we are simply adding the soil in the extraction plate. So for single line culture, when the nematode came at the center, uh, we used to pick that nematode with the help of nichrome loop and transfer it to the another fresh agar plate, plain agar plate, and we used to supply bacterial suspension. So we use E. coli or same soil supernatant for the food feed. And after now for the single line we use the this not the soil supernatant. We use E. coli suspension. So after the nematodes grow on the single line culture, so the single nematode is transferred on that. So what happened there? The single nematode gave eggs, and that plate will contain only the single line of that nematode. So this how we used to make the single line in the of the nematode in the lab. So there was no sterilization technique in the lab itself also.
Yeah.
Just a second, I have a question to Mandar. So you just talked about the creating a single line. So can you more elaborate why do we need single lines?
Okay, Shivan. So what was happening? If we do any of the experiment like olfactory assay or anything, so if we don't have a single line, what you will compare with? So if there will be two different nematodes and we are trying to do an experiment, so that will be of one nematode will be of different species and another nematode will be of different species. So if we identify a single line, we can go for an identification like what species of nematode it can be. For identification, even we are trying to identify, if we obtain a single line from soil nematode, we go for identification that what species it can be, what genus it is. So last year, I think so, Kunal has found Ostrich tipole, Nasino Rabdopushanli.
So
Yes,
one
yes.
question
Yes.
from YouTube is, if there are two nematodes of the same species, will you still call it single line, and will it do the same work?
No, basically, so what we do is maintaining the single line culture. We mean to have a pure culture of only one species. So there is a possibility that the other species of nematode, the egg of the other species of nematode would get carried along with the single nematode which we are picking from the extraction plate and keeping it on the SLC plate. That is a single line culture plate. So there is a possibility of that. And so we have to keep making SLC, more and more SLC, so that we can be sure that the culture we have is of the single species only.
So again, there is a question: why single line culture? If there
Saida,
is,
I think
okay, we can take the more
we have little
questions.
time limit. Saida, is there important like repetition or another question?
It's a similar type of question. We can take that later, maybe.
No, not
WhatsApp
later. You can put it in our WhatsApp group, and we can discuss them.
Yeah, I think there are some participants on YouTube also. So when you are viewing this, if you have any questions, so you can get in touch with us with the links in the description.
And the presenters also can reply if possible to the YouTubers. YouTube.
Yes.
Directly they can write.
So we have some
Can
links.
you just in 1 minute can you talk little bit about context to curriculum also, what you are trying to do with your study?
Yes, that's important.
And also like you were talking about comparison, so you can take little bit, half a minute, you can talk about what you are comparing between lab bred and native.
Yeah, and meanwhile, the next session, I think the group should get ready.
Yes,
All the moderators, please ensure that we are ready for the smooth transition into it. Yeah. So over to context to curriculum discussion of nematodes. Jay, thanks.
Viewers, you can continue your group.
Yeah. Ramdeepak, can you move to the concept map?
And you can you can say about your long-term objective, like you are talking about comparison, right? So you can tell about that.
So talking about comparison, what we were planning to do is like we have a standard strain which is known as N2 strain, which is a lab bred strain. The lab bred in the sense a big term, so I will simplify it. It's called, we generally use that particular thing in the in a lab, in normal doing any research, any answering any research question. We want to find out the native strain of the C. elegans, and we want to see that is there any effect of domestication occurred or happened. Domestication is what, like we are breeding the we are culturing the nematode in the same media, in the same with the same amount of feed. We are all giving it in a restricted proportion, whereas a native strain which is present entirely on the nature, it's like it's facing a lot of stresses. So is there any really a difference between both the things? We are going to compare this with the help of the olfactory assay. That's our long-term objective. And if we find the C. elegans from the Indian native soil.
So you are going to do olfactory studies with this.
Yes.
With the native, comparing the olfactory study of the lab bred.
Yes. So we can do several type of studies, but currently focusing on one single study. That's the olfactory assay.
Okay, fine. And you want to talk about this context to curriculum? Anything? You have only 5 seconds.
Yeah, okay. Just 5 seconds. Okay. So, concept to curricular things, what we have tried to make out is like we have studied all this type of subjects in our undergrad studies. So we just try to relate like what we did in our lab. So research methodology, first point is one of the very much important point because we talk about searching of research papers and also getting to know how like how important the model organism is. So we have mentioned that over here there's a history of nematodes or C. elegans, how it evolved, and also finding relevant research papers. So a research methodology. Talking about biostatistic, because when we do any olfactory assay or olfactory study, we need to be very clear about what is the expectation of what are the results we are expecting for a particular study. For that, we need to have an idea of how the statistically important the data is. So maybe how, like what is the expected result. For that we do a population density study, like how much nematodes are moving towards a particular concentration and why the nematodes are, how many nematodes are not moving to a particular concentration. So we have included biostatistics. Developmental biology, because we focus on the life cycle of nematodes, the different larval stages, the embryonics. We have also during when we are working in the cube lab, we tried to find out like the developmental stages. So we have found around from 2-cell stage to 4-cell stage in the cube lab, and the picture was also there. So focus on a developmental study. And also one of the important or like interesting fact about nematode as a whole is like they undergo a DOR state. So DOR state is a dormant stage. So if they face any stresses, so they undergo DOR state. So we are also interested to study about DOR stage. So from the DOR stage we also can get to know what is the importance of DOR stage in a particular, because they get into a DOR stage at the larva 2 level. So what is the importance in the developmental biology? So talking about the neurobiology, what we mentioned about, because we are doing olfactory assays, so there are like maybe connecting neurons, and also like evolutionary studies. We talk about mutation, natural selection. Niche biology, because we are talking about comparing both the strains from the lab bred as well as the native strains. So our niche biology, different soils, like we can also compare like whether what is the difference between the C. elegans found or the nematodes found in different soils. So we have connected to geology. Taxonomy, because we have successfully identified in this region about the Caenorhabditis genus. We have found Caenorhabditis genus. We have like narrowed it down to the genus level. We have successfully identified that Osseous stipulae with the proper reference, and we are hopefully trying to find out C. elegans. Then talking about reproductive biology, why we mention reproductive biology, because we are maintaining a single line culture where we say that a single mother is important to maintain the single line culture. So we need to know about the reproductive patterns and how many eggs it's going to lay after particular days. Then there are many of studies which is done on ecological interaction as well.
Yeah, Bhivas, I think there are, like you have written about genetics, cell biology, molecular biology, because all these things are connected with your work, right?
Yes.
So, and it is a context for even studying and about the topics of the curriculum. Okay.
Yes.
So yeah, we have a little bit limitation of time. So I think you can also discuss on the WhatsApp group. We have a lot of more questions, which can be discussed on we are posting on the WhatsApp group. So you can you also should participate and discuss on to those questions in the group. Okay.
Yes, sure.
Okay, so shall we move to next presentation?
Yeah. There's a question from Arundan sir. How hopeful are you on getting C. elegans? I think the hopefulness started 2 years back and it's still continuing. So why not be a little bit more hopeful and let's be the first person to find out C. elegans from India.
Okay, so I would like to now let's move to next presentation, which is the next one. It is a snail presentation. So snail group is ready. All the members of the snail group are ready. So so please snail group. We will be calling you for the next presentation. It is a study on learning and memory in the snail model system. So learning and memory is also very interesting area in the biology, because it's also one of the Nobel Prize area where the Nobel Prize has gone in. In the starting of this century only, to the year 2000. So it's a very, very frontier area, and they are studying using a snail model in their home lab. So they are showing that even the complicated stuff like Nobel Prize winning work can be done with a simple system in the home lab. So I would like to call Snail Group for the next presentation. Snail Group, please present your. You can start with your presentation.
Yeah, Kunal, I think you can take over the presenter and set things up.
Good evening, everyone. Today we are going to learn some things about very giant snails, which are called.
Shall I start the video?
Yes, Kunal, you need to unmute
Yeah,
it
please.
and you can start. Yeah, the snails are silent, so there is no sound in the beginning, but we'll hopefully have some soon. It's a silent beginning.
For the maintenance of snails, we place the snails in the tray that can be shown in the picture, and we fill the tray with a little bit of water, and we place food such as carrots, cucumbers, and cabbages. For why we place water is because the snail's body shouldn't dry up, as their body is slimy. So, and without water, it can dry up. So that's why we do add water. After the snail is done feeding, I place the snails in the basket. The basket and the lid, which is this part, they are together and they are not separate. After they are done feeding, I place them in the basket and I slightly fill the basket with water so that the snail's body do not dry up, and I close the basket so that the snails do not escape while I'm not watching them, and that's it.
Giant African snails eat a variety of things, which includes leaves, fruits, vegetables, tissues, paper, and organic matter in the soil. According to the design of our experiment, we feed the snails with coriander and cucumber. Each snail is provided about 4 to 5 leaves of coriander in a time interval of 24 hours. Cucumber slices of moderate size can also be provided to the snails. The food is kept in petri dish, which is then kept inside the container in which the snails are kept. In addition to that, chalk powder is provided once in a week as a calcium supplement. Every day, the leftover food is
Olfactory assay. Why to perform olfactory assay? Olfactory assay is done to observe the olfactory ability. Olfaction is defined as the detection of environmental chemicals present. Olfaction is the sensory principle. Therefore, olfaction is done to see the behavioral change in the snails after we provide it with the coriander for a period of time. We feed the snail with coriander and starve it for 24 hours before the assay.
Tank should be covered with the black paper, but we should not cover the one part of the tank because we need to observe where the snail is moving.
Why to perform control versus control assay? Because we need to calibrate the instrument, that is, we need to check whether there is any odor or smell of any molecule in our setup, that is the olfactometer.
Steps to perform control versus control assay.
First, take blank white paper, then draw a circle of diameter 10 cm and a radius of 5 cm. Second, then draw a perpendicular line in between the circle, then mark the direction, that is the north and south pole, and C1 and C2 at the either side of the perpendicular line on the circle. Third, then place a glass slide on the paper and with the help of the dropper, put a drop of distilled water on C1 and C2 point. Fourth, then place the snail at the north direction and then cover the slide with the tank. As soon as we cover the slide with the tank, we should set a timer. Fifth, like this we should take 20 readings, and for the rest of the readings the snails should be kept alternatively, like for the first reading at north direction, then for the second reading south direction, and so on. And after all the readings have done, we need to calculate the ORI, that is olfactory response index. The formula for ORI is C1 minus C2 upon C1 plus C2.
Then we perform olfactory assay with sample, that is the coriander juice and the distilled water. The procedure is same as that of the distilled water versus distilled water, only we put in C1 side distilled water and C2 side coriander juice. We observe that the snails move most of the times towards the coriander juice, that is the C2 side. That is, out of the 20 readings, almost 17 to 18 times the snail moves towards the coriander juice, that is the
This is Eric Kandel, and he studied nervous system in sea slug. His breakthrough came in 1970, and he was subsequently rewarded the Nobel Prize in 2000. So I'll be talking about Eric Kandel's experiment, and what his experiment basically was that it was based on learning and memory, wherein he made the slug to not react to its touches, or in simple words, habituate the slug, and then he resensitized it by giving a small shock to its gills, so its defensive systems are reactivated and it is resensitized. So I recreate the same experiment with my snails. So you might ask why I'm using my snails. So the reason why I'm using my snails is because the tentacles are somewhat similar to the gills that are shown in Eric Kandel's experiment, the gills of the sea slugs. And the sea slug's gills and my Achatina filica's tentacles, they both withdraw or retract on stimulus. So I'll be talking about my Eric Kandel's experiment recreated with my snails. So as you can see in this, I touch one tentacle and both of them retract. Both of the tentacles retract. This is normal, because if someone tries to touch your left eyes, you will naturally retract, or I'm sorry, not retract. You'll close both of your eyes. So this is natural. And later on, when the tentacles start to come up again, it is to be noted that the tentacle that I've touched has come a little later than the tentacle that I have not touched. In this part, I touched the left tentacle which was previously touched, and instead of both of them retracting, only the left one retracts fully while the right one has been shown stable. Again, the left tentacle is retracting fully while the right one is unfazed. And in this one, I touch the right tentacle, and it looks like the right tentacle does not the right tentacle contracts while the left tentacle does not contract. And now I resensitize the snail by giving it a strong stimulus so that it forgets the habituation that had been learned earlier by the snail. So you can see it from now onwards. Here I give a strong stimulus. It makes both of the tentacles retract. And now when I touch one tentacle, both of the tentacles withdraw.
Hello everyone, I am Firoz Patel from Makerspace, collaborating with Cube Snail Group. In this video, we can do a simple exercise in which we can control a motor with a sensor. Here, this sensor is a sensory nerve in the siphon of the snail, and the motor is the muscles of the gills. And Arduino is the internervous of the snail. So let's do it. When I move paper closer to the sensor, motor starts or muscles start moving. When I remove the paper, the muscle stops moving. Let's do it one more time. When I put it, it starts moving. Remove, stops. Let's do it for more time. Let's put the paper for more time. After some time passes away, the snail habitual of the touch, so the muscle stops moving or motor stop rotating. That's all for this video. Thank you.
Why this sudden reaction? Reflex arc is a neurological and sensory mechanism that controls a reflex action. The receptors on the hand skin receives a stimulus and then transfers the stimulus all the way to spinal cord through a sensory neuron. There, another relay neuron passes the signal to the motor neuron. The motor neuron transfers the signal to the effector muscle and the muscle contracts.
Abnormal learning and memory retention abilities can lead to several diseases in humans. One such disorder, which is linked to learning and memory, is Alzheimer's disease. What we learn is stored in specific areas of the neurons called the synapses, which are the areas between two neurons where they communicate and information is passed in the brain. Sometimes in the brain there is accumulation of beta amyloid proteins which are released along with neurotransmitters. These beta amyloid proteins accumulate and block the synapses, which makes the person unable to remember things and difficulty in carrying out day-to-day functions. Other hypothesis is that the tau protein, which is responsible for holding the microtubules, begin to pair with other tau proteins. As a result, the microtubules disintegrate and tangles are formed inside the neuron. This can result in the malfunction or death of the neuron. This leads to the shrinkage of the brain.
Hello doctor, thank you for taking off time from your busy schedule. Say, how can I help you? I need to submit a project on Parkinson's disease and I don't know what a person goes through in this disease. Can you tell me about it? I can try narrating a story of one of my patients so that you get a broader view of what a person goes through in Parkinson's disease.
He had become a lizard, a great lizard frozen in dark, cold, strange world. His name was Robert. He was a tall, thin man in his 60s, but like most patients with Parkinson's disease, he appeared to be much older than his actual age. Not many years ago, he has been active, vigorous businessman. Then it happened, not at all at once, not suddenly, but slowly. Now he turned like a piece of granite, walked in slow, shuffling steps, and spoke in a monotonous whisper.
What has been his symptoms? A tremor. Had his tumor been disabling? No, his hand tremored the worst when they were doing nothing at all. A symptom called tremor at rest.
The other symptoms of Parkinson's disease are not quite so benign. They can change a vigorous man into a lizard. This includes rigid muscles, a marked paucity of spontaneous movement, difficult in starting to move, and slowness in executing voluntary movements once they have been initiated. The term reptilian stare is often used to describe this characteristic: lack of blinking and the widely opened eyes gazing out of a motionless face, a set of features that seem more reptilian than human. Truly a lizard in the eyes of the world. A small group of nerve cells called as substantia nigra, black substance, were uncountably dying. These neurons make a particular chemical called dopamine, which they deliver to another part of the brain known as striatum. As the cells of the substantia nigra die, the amount of dopamine they can deliver goes down. The striatum helps control movement, and to do that normally, it needs dopamine.
Thank you for the explanation, doctor. Happy to help. Bye.
So I hope you all liked this video and have got some idea about what we are working on. So if you have some questions, you can ask now, and we'll get to all the questions in the public chat right after that.
Yes, question.
So the video was amazing, but so many people have confusion that why you are using the coriander only.
Okay.
Reading this.
I would like to add to that. It's like why I asked this question. So you're using coriander for olfactory assay. So if you're using coriander for olfactory assay, considering it has a strong smell, so will you be able to create the gradient that you need for the olfactory assay?
What do you mean by ingredient? Like an ingredient that imparts the smell?
Yeah, gradient. Yeah, we are able to create the gradients by by using coriander. We mean that we are using coriander juice. We extract coriander juice from coriander by crushing the leaves and taking the juice out with the help of a muslin cloth, and then the juice is then diluted with the help of water, distilled water. So in this way, we are able to create gradients of coriander.
So is it like decreasing the sense of smell out after you're dipping it or diluting it in the water, and which what concentration you're using for the coriander juice?
As the tank is not that big, so we need if we use the concentrated one, the tank is not that big that the smell will not spread in the entire box. So first we do the concentrated one. After that, we start dilutions like 10 raised to 1, 10 raised to minus 1, 10 raised to minus 2, to see if the snails exactly go towards the coriander.
I think, Nishmata, we should elaborate what is 10 raised to minus 1, 10 raised to minus 2, because people... Yes.
Hello. Also, there was the question raised by Lokesh, why are you specifically using coriander? So that was unanswered.
Yes, also if we can use something other, because considering and what are your expectations for a particular result? Like when you're using a total concentrated one, as someone said, so what are you expecting out of that particular assay?
Okay, so what Lokesh had asked us that
also.
Okay, no, please go ahead.
Also, if the coriander is not available, it was someone asking on YouTube that if coriander is not available, any other food you can provide snails.
So we generally use cucumber, tomatoes, as you've seen in the video as well. It's not very particular to coriander only. But when we are doing olfactory assay, especially we don't say it is completely olfactory assay, it is learning and memory. For that we need one same food throughout. So if you are using tomato or some any other food, you have to continue that food. So I think that would be elaborated more because it's not just checking their olfaction. It is we're trying to study learning and memory, right? So we need same food throughout. So it's not just coriander. You can add anything that is available at home. Like you can use
As
cucumber.
Yash has said, it's like coriander only. Then if snails like coriander, who feeds the coriander in the wild? So how they get from in the wild?
So snails
Actually, snails eat a lot of foodstuffs, so it's upon us that what food we have to give to them for our experiment. It's like we designed the experiment, so we have the freedom of choice that what do we want to feed the snails. So there are options like tomatoes, cucumber, and coriander. So from that we are choosing coriander because it has some good smell. So that is it, and so we have got the freedom to choose. So we have chosen coriander. So only thing is that we stick to that food stuff. Like we give coriander only to the snails for months to come during the experiment, or else if we are choosing cucumber, then we can stick to it and give it for longer durations, like, as our experiment demands. So yeah, there's no issue like what to feed the snails, but it's upon you, your decision.
And
Yeah.
also you talked about okay.
Yes.
Arunima, go ahead.
So what will be the perfect environment for the snails live?
The perfect environment for a snail is moist environment with and it should not have direct sunlight, as snails are more active during the night. So like they prefer like humid climates and dark conditions.
So
How light
Anjali,
how, how light
yeah, we
affects the snails?
Okay, so I'll circle back to Lokesh's question that he had asked before, that what about why only we choose coriander, but I thought Bivas cleared that, but we choose coriander. I mean, we can choose other foods like cucumber, but we choose coriander mostly because it has a strong and distinct smell, and coriander is like easily available. So and yeah, so we choose coriander for that reason. And can I get another question? I
Yeah.
guess
So
yeah.
my question is, like, you talked about memory. So like how you describe memory, like how you describe memory and how you're connecting the study of olfaction that you're doing with the Parkinson disease and Alzheimer disease. I think that is one of the question of Jay sir as well in the chat.
Yes.
Memory is something like learning is the change in behavior, and memory is that retention of that change behavior. Means we use that means we use that change behavior, and we can relate.
So
Yeah, so what in the experiment, yeah, I'll connect it that in the experiment what we are doing is like we have we all it fact that snails are eating coriander, that is a natural fact. So that is their natural behavior and we prove it with the help of assays. So but we bring a change in the behavior of the snails by feeding them with coriander that is mixed with quinine. So once the snails get to know that now the coriander is having or giving bitter taste, so then in the next assay they won't go towards coriander. So this is the change in the behavior of the snails. First, the same snail used to go towards coriander to eat it, but now after the conditioning, they
Mansi, my question also is that how do you connect it with Alzheimer's and Parkinson? So this memory thing,
Yes.
how are you connecting? Because you talked about Parkinson, Alzheimer, and all. But how that experimentally, how are you connecting it with the Parkinson, your snail experiment?
Yeah. So when the snails learn and when they memorize something, so there are connections. Means the cellular form of memory is a neuron. Neuron is situated in the brain and yeah, also in other parts, and it is responsible for responses and for feeding the information and memories. So what happens is when we learn something or when we learn something, then there are synapses or the connections between two neurons which get increased, and in Alzheimer's what is happening, these connections are getting blocked. So is there any way so that we can re-establish the connection between these neurons? So if we figure out a way to do this in the snails, I think that we can also imply that in humans, or at least try to do so.
Mansi, you spoke about like increasing of synapses. Can you like more elaborate, like what do you mean by increase of synapses between two neurons?
Or are you saying that there's some pathway, like molecular pathway? Like you saw
Yes.
that this thing also, no? The mechanical thing which from Maker's lab, the experiment was there, in which they were showing that habituation, right? That it was moving, right? It was a simulation of snail experiment, right? In the Maker's lab also.
Yeah, we could ask
Yeah.
Firoz to speak. He's, I think, right here. Can
Yeah. Who had done that? They can speak. Yeah.
Yes, Firoz.
You can say.
Yes, sir. So what is question, sir, please?
So you actually made a simulation of snail experiment into your with your machine, right? With your device in the Maker's
Yes.
lab. So can you just say like what were you trying to say? Like how did you make it actually, and what gave you this idea?
So I use a microcontroller which is Arduino. So when a sensor use is a when anything touch the sensor. So when it's the thing time. So the motor stops moving. Relation is iron.
Okay, so I think what he's trying to say is when you touch the sensor, the motor stops moving. And he's doing it using some microcomputer called Arduino. Am I correct? Firoz?
Yeah, sir. Yes, sir.
Okay.
So it is something like that there is a pathway going on, and then there is a disturbance, and then that pathway stops, right? So same way your reflex action also is the same pathway, right? So there's some problem, then the pathway also stops. Or if there is a Parkinson, the pathway can stop like that. I'm just making a guess.
Yeah.
Yes. Arunima is asking how habituation is achieved. Can you answer it, Anjali?
Yeah. So we achieve habituation by by touching. I'll just explain habituation clearly. So habituation is like the delay in response, or like the organism stops responding to stimulus given to it, given to any part of its body. So this is habituation. So how we achieve a habituation is, for example, we touch the left tentacle of the snail, and it has two tentacles on top. It's the left tentacle and right tentacle. So we touch the left tentacle for the first time, and both of these tentacles retract. Why both of them retract? It's basically the reflex arc of the snail. It's like basically the reflex response of the snail. So both of the tentacles retract. So, and after that, after the second or third touch, I touch after the third touch, the left tentacle on touch retracts, but the right one does not retract. So it's slowly figuring that what I'm doing is not harming the organism at all.
So what is your hypothesis? Like what changes may be happening when that right tentacle is not withdrawing, for example?
Okay, so what I hypothesize is that the right tentacle is slowly realizing that what I'm
No,
doing
what do you
is
mean by realizing? What do you mean by
okay.
what chemical changes may be taking place as according to you, so that it is now realizing that it doesn't have to withdraw. So what kind of molecular changes do you hypothesize may be happening in that small period of time? Like it's just a fraction of seconds, right? In that
Yeah.
fraction of second, there is some change happening.
Okay. So the receptors, sorry, the neuron relays, and the neurons that relay the touch of the touch of my stimulus to um, uh, the brain of the snail. So they slowly reduce. So for example, there are CA calcium receptors present on the motor neuron, and the motor neuron basically it relays the stimulus that I've given to the brain, so that the brain can give further information on what to do. And in this example, the brain informs the snail to withdraw its tentacle. Okay.
There's a memory form, like we were saying about memory, as in
Yes, yes. Is it a form
So
of
how is memory?
forming
How is
synapses?
memory formed? How is memory formed in the
so it is
tentacle?
like associative memory. So as in when we talk about quinine and coriander, the snail was going towards the coriander each time, but when we added quinine, it stopped going towards it. It is instead going in opposite direction. Um, why is that? Because I think so we hypothesize it that it is kind of a associative memory where the snail is associating the coriander now with quinine with a bitter taste. So the exact pathway is we have still not studied that, but yeah, the
I have a question.
yes,
Yeah, please ask.
Hello.
Hello.
Hello. I have a question.
Yes,
Yes, please answer.
Yeah. The question is, how many snail in this planet?
That's a really difficult question to answer
You
because
can count yours. You can count.
How can this question be answered? Like,
it's
Yeah, that
same as
answer
asking like
cannot be
how many
given by
ants.
any human, I guess.
Yeah.
You can do a census of snail.
you can track
Yeah.
random snails in your place, but in the whole world that would be difficult.
But I can say that snails are a pest, and that's why there is so much of like that is also a reason so much of studies going on. Actually, snails feed on like literally anything, like plants, fruits, or organic matter in the soil or small soil particles. So what is and they also have a high reproduction rate. So the population
I
they
have one more
start
question.
growing. Yeah.
Yeah, the maximum and minimum height of a snail.
Yeah, actually, there are snails of many species. Like there are small snails, there are huge snails. The snails which we
Okay,
are working on.
Mansi, can we come to like
Minimum
other
and
questions?
maximum. Maximum कितना और minimum कितना है वो?
No. See, Aryan, see, why that question? We have to also think that why we are asking that question, right? So, why it is important to know exactly how many snails are there, right? So, we are discussing something on research, right? What actually can be done with the snail, not how many snail do we have. So,
There is a comment from
is there any other
Arunan
question, or
sir.
we can move to our next
There is a-
presentation because there's a
There's a
limitation
comment
of
from
time.
Arunan sir. There's a comment from Arunan sir. Arunan sir is asking, saying it's like tentacle withdrawal cannot be associative. Like Mansi and Anjani, can you elaborate?
Can you elaborate the question because I didn't really
Yeah,
get
that is not
Statement
clear.
from, there is a statement from Arunan sir. Tentacle withdrawal cannot be associative.
So I think I think what Anjani was trying to say is when he touches the tentacle of the snail. First time it retracts, second time it would retract, but like if you do it continuously, it will not withdraw the tentacle. So I think there would be an associative memory from there as well, where the touch is maybe I'm giving it a humanizing term, but right now it's not harsh. It's a sensitive touch. It will not harm the tentacle of the snail, and maybe that's why it's not retracted.
So how can you say that it is not harming the tentacle of the snail? And considering if I if you give you from the broader perspective, if someone pricks us with a pin, the number of time the person pricks me with pin, I will or retract my hand. So
wait. To this device, if someone someone pinches you, or someone picks your finger for the first time, there would be a sudden reaction. But when it continues, when someone is doing continuously, you know it, right? You realize it, that okay, this is going on, and because you are a human and you can speak and you can talk, you'll of course say what's wrong with you, why are you doing that. First time you might not behave the certain way, similarly like touching at the
but it will be always a retract manner. So I was asking when
retract manner. See, okay, so
Sweta, I had a question.
Yes.
Excuse me, I'm sorry to intervene, but I just needed to ask. We are talking of olfactory assay, and we are touching the tentacles. So, can you please elaborate and explain?
There are two different experiments as of right now, like olfaction and touching of the tentacle. Yes. As in, it's the same study, but two different methods are used. So yeah, it is, the touching of the tentacle is not related to sense of smell, that is olfaction. But yeah. So
basically, you're teaching us a learning and memory.
Yeah.
Okay, so you're training the snail. Thank you.
Yeah.
So Arunima is asking how we can connect the snails and arachnid sea slugs.
So sea slugs
Okay.
and yes, Anjani.
Yeah. So Arachnid sea slugs. Arachnid sea slugs, they had gills, and on touch, on stimulus, they retract. So it's similar to my snail, like our snail. So and our snails, the tentacles also on touch, they retract. For example, like the Arachnid slugs, the gills contract, which is like very similar to the snail's tentacles retracting. And when Arachnid touch these gills of the slugs a lot of times, so that's why so they stop retracting. And similar to my experiment, when I touch the tentacles of the snails a lot of times, they stop reacting. So in that way, we can relate that Arachnid sea slugs are very similar to my snails.
So you are saying that it is similar. Your experiment is similar to Arachnid.
Yes.
Yes.
Sort of.
Yeah, but so you are also studying the same thing what Arachnid actually was studying in the sea slugs. You are using a snail as a model because you don't have slug. That's why you are using snail.
Yes.
And so my only I think we should go ahead a little fast. That what experiment you can do to see what is actually happening inside neuron when this change is happening, when habituation is happening. How will you track that change, or how will you see in the neurons what changes are happening? Can you do some experiments?
Okay, so to see what changes are happening in this in the inside of the snail, we'll have to delve a little more into molecular biology. So
Can you
so
tell me in a 5 seconds? Yeah.
So that should be a future plan, but for example,
I hypothesize that as memory is formed, there are modifications in the neuron, like more synapses are formed. So I think that there will be an increase in the protein content of the neuron. So if we figure out a way to estimate the protein the amount of protein in the control snails and the ones which have a new memory form. Then if there is a difference, then we can like kind of prove our hypothesis.
Yeah, so you can trace
Mansi,
down
a
some
quick
of the
question.
proteins from
What is
from
the
the cell.
protein control you said, like which amount? What protein you're talking about, like protein content in which sense, like inside the neuron or outside the neuron, in which sense you're talking about the increase in the protein content?
It's about the synapses and neurotransmitters.
So in the tentacle cells, are you saying that?
Oh, I'm saying it about the neuron.
Yeah, neurons in the tentacles. Neurons where?
Yeah, neurons connected to the tentacles and the brain of these snails. So because they basically relay the signals to
Yeah.
the
Okay, that's
brain.
fine. So we can discuss on the group about how we actually exactly will do that. Okay.
Yes.
So I think we should move. We don't have time, so we can now move. I think we have discussed a lot of things. We can now move. Any other question is there? We can put it on our WhatsApp and our groups and all. You can also write on our STEM game and all. You can create some thread. We can discuss there. Okay. So let's move to the next presentation now.
Yes, but before that, I think because we are at the middle of the session, I think we have a lot of participants here. We can take a group picture, so and people also can relax a little, because I think we had really intense 2 sessions with really big goof up in the beginning with the technical thing. So let's celebrate that with a small group photo. So ideas on how to do that? Jen, you want to suggest something?
We do have option for share webcam.
We can,
Is it the same as the opening?
You can share webcam and take a screenshot of
If you
it.
share the webcam, then automatically the screen will adjust.
Okay.
And will come in. Yeah.
Okay.
As much as possible, all of you can join. Yeah.
Yeah. So let's let all of us turn on the cameras. So it's the third one on your screen. So I will do it. But I think there might be some technical thing. If let's try. So all of us, please turn on the cameras. Yes, I see Ashutosh. I see Sumita, Prashant, Vivas. Yes, we need more students. Please turn on your cameras. Oh, this is wonderful. It's adjusting on its own. So maybe I think I will have to count 10 for all of us to join. So 10 seconds, 10, 9, all of you please turn on cameras. Jay, Ravi, Ramdeep, Arunan. 7, 6, 5, 4. I see only 4 into 3, 12 people. Yeah.
I'm
Thanks.
getting the option as webcam locked or something.
We need to drag Arunan into the screen.
Yes, Arunan.
I heard a woof. Some dog also wanted to go.
Yes, it's from my side.
Yes. So he also needs to be there.
It's showing logged. Webcam sharing is logged.
I'm unable to
I
share.
am also
my-
unable. It is logged.
Yeah, but
you have to give access to camera.
that you are
I
unable
gave-
to is also being recorded.
I gave access, but let me try.
You may have to give the browser access. Yeah, that's right. Yeah, in case you haven't used it before, you have to do that first.
think it's long.
Yeah, meanwhile, I think we will also prepare for the next session.
Yeah, this is like a trial group photo, and be ready to take more group photos tomorrow and day after, and also on Saturday and
Yeah.
Sunday.
Yeah, I have taken screenshots, quite a few.
Yeah. And Arunan is missing.
Arunam sir is not there.
Yeah.
Probably because many people have on their webcams. Is it that the others can't join in?
No, it's not the reason. No, you just have to. Each one has to put. I don't think everybody actually has webcam. Maybe some people are hesitating to put on the webcam.
Unable to do. So not logged in.
Firoz and Farhan, you can also join. Turn on your webcams. You can. Ashish. Yes. Raj Yashwant. Rahil. I think this is the maximum number with 14 people.
So we can have a screenshot of this and then others can join.
I think we can disable the privacy or participants.
Yeah, that I have minimized myself. You are muted, GM. Yes, so we have now 15 people.
Where is Kiran?
Yes, Kiran.
Kiran is not in this room now. As in Cube Chat Shala.
No,
She's here. She's here.
we have to drag her inside. She has no
measure it with oscilloscope.
Yeah, I think that's a discussion that we can have. Yes. So that's
Hey everyone, my project is a smartphone as an oscilloscope used for home lab. It is a very crucial instrument in the field of electronics. It also used in bio. This yellow color stuff is PCB. It is a combination of electronics component which I use while making an oscilloscope. In the second figure, I use a smartphone and a display. If you are interested in making oscilloscope, you need these components. This is the audio jack. We all human can use this audio jack for listening a song, recording, etc. Here I use this audio jack for sending a signal to my phone. This is a circuit diagram which show the actual wiring connection being used. If you are interested, I will explain this later. I have captured some predefined signal because I need to check whether it works properly or not. Some examples are sinusoidal wave, triangular wave, square wave. This oscilloscope has specified the voltage range from 10 mV to 50 volt. The frequency range is also limited from 75 Hz to 1500 Hz, but it does not capture any DC signal. Application of an oscilloscope. Oscilloscopes to detect heart irregulation. It also used in nerve signal measurement. It is used to diagnose the brain signals.
Thank you for listening.
Yeah, so was there any audio issue? I saw some messages saying that video was not playing and audio was lost. Is it so with everyone? Okay,
No.
okay, you could hear. Yeah, so that was a very short and quick video that Farhan could make. So unlike many of the model systems, so currently we just have one student.
Hi, sir. Hi.
Farhan. Yes, you can speak and. So can we, Kiran, also upload the slides if we have for the oscilloscope when we speak? Yeah, thanks. Yes, so Farhan, like, what is this oscilloscope, and let us see if the students have any questions looking at the quick video. I'm pretty sure there will be a lot of questions like, "Ye kya hai? What is this going to do?" And, right? Please feel free to ask, and let's try to figure out.
Farhan,
Yeah.
what
Yes.
is I mean, how can we use this oscilloscope in like I am a biology teacher, so how can I use this oscilloscope in my subject? Like you said, we can calculate heart rate. Can you
Yes,
please elaborate on that?
Yeah. Yes, ma'am. Hello.
Yeah, Farhan, बोलो बोलो.
या यस मैम, ऑस्लोस्कोप को हम इस तरह यूज कर सकते हैं कि जैसे हम जैसे हमारे फिंगर के पास एक टिप करके एक सेंसर्स होते हैं. सेंसर जनरली नॉर्मल जो यूज होता है वो आईआर सेंसर यूज होता है. आईआर सेंसर पे एक ओपेम्प लगा करके आउटपुट लेते हैं, और उसमें जब मैंने जो ऑस्लोस्कोप बनाया ना, उसमें 2 प्रोब है, मतलब क्रोकोडाइल क्लिप है. तो एक पॉजिटिव है, दूसरा ग्राउंड है. तो उसको वहां कनेक्ट कर देंगे ना मैम, तो वो सिग्नल जो है मेरे सर्किट के थ्रू पास होगा, और जो मेरा मोबाइल फोन है ना, उसमें वो सिग्नल डिस्प्ले होगा कि पल्स रेट कैसे, मतलब हार्ट बीट कैसे वो फ्लक्चुएट कर रहा है, चेंज हो रहा है. तो वो इस तरह हम देख सकते हैं.
Saida, can you allow multi-user? Multi-user, yes. Yes, please, Hina, continue.
या फरहान सर, एक चीज ये बताइए, जैसे आपने हार्ट रेट के लिए बोला, वैसे ही जैसे कोई स्ट्रेस में है, कोई गेम खेल के आते हैं बच्चे, और हम लोग अक्सर उनका स्ट्रेस लेवल चेक करते हैं। स्ट्रेस मतलब बॉडी का ब्लड प्रेशर बढ़ा होता है, पसीना आ रहा होता है, तो
Yes,
हम
ma'am.
क्या वो भी डिटेक्ट कर सकते हैं? सांसें उनकी तेज चल रही हैं, तो क्या
Yes,
कैन वी जस्ट हम लोग इसको मॉडिफाई कर सकते हैं? उनकी ब्रेथ रेट को भी देख सकते हैं पल्स के साथ साथ?
यस मैम, देख सकते हैं। उसके लिए बस क्या करना होगा, जो इंटरफेसिंग है ना उसका, मतलब ह्यूमन बॉडी के साथ जो हमें मतलब वो क्या, वो सॉरी ब्रेथ का आपने क्या कहा मैम?
ब्लड
जी
प्रेशर
जी,
और
सांसें सांसें।
हाँ सॉरी, हाँ
हाँ जी,
तो
सांसें।
उसके लिए जो मतलब इंटरफेसिंग जब बॉडी से करनी है ना, उसका बस वो करना होगा। और जो उसके जो मतलब प्रोब्स है ना आउटपुट, उसको बस इसके ऑसिलोस्कोप से कनेक्ट कर देना होंगे, तो वो हम उसको भी देख सकते हैं मैम।
ओके, इसका फरहान कॉस्ट क्या आता है? व्हाट इज द, इसका लागत क्या आता है बनाने में?
यस मैम।
जैसे अगर बच्चे ये प्रोजेक्ट बनाएं और उसका एप्लीकेशन हम लोग डिस्कस करके इसको अगर हम एज अ मॉडल बनाए घर स्कूल में, तो क्या कॉस्ट आता है इसका?
अप्रोक्स 60 रुपीज ऑन लिया था मैम।
60
तो
रुपीज।
फॉर 70 रुपीज।
ये बोर्ड जो है वो आपको मिल जाता है क्या ऑलरेडी बना बनाया? मतलब ये जो बोर्ड आपने यूज किया, आईसी वो सब यूज किया आपने, शायद रेजिस्टर कुछ यूज किया है, ऐसा कुछ?
नो नो, ये मैंने खुद डिजाइन, मतलब सर्किट डिजाइन किए, मैंने इस बोर्ड को
आपने
बनाया
ब्रेड
है खुद
बॉक्स
से।
ले करके बनाया होगा ना?
या
हाँ, तो ये सर्किट आपने किस तरीके से बनाया, ये मतलब कोई बता सकते हैं? कोई बना कैसे सकता है इसको, बिल्ड कैसे कर सकता है?
या यस, आप इसके लिए बहुत सारे फ्री सॉफ्टवेयर्स हैं जिसको आप डाउनलोड कर सकते हैं। उसके थ्रू आप उसमें सर्किट डिजाइन करते हैं, सर्किट डिजाइन करके आप इसको
मिनिमम
फिर
चीजें क्या लग सकती है, मतलब मिनिमम चीजें क्या क्या चीजें उसमें मेरे को रिक्वायरमेंट खरीदना क्या क्या पड़ेगा?
इसमें आपको मिनिमम रिक्वायरमेंट होगा, पीसीबी लेनी होगी, एक नॉर्मल कॉपर पीसीबी, और आपको एक
व्हाट इज पीसीबी? बिकॉज़ यहाँ
पीसीबी होता है, पीसीबी होता है प्रिंटेड सर्किट बोर्ड। वो एक नॉर्मल जो हम कार्डबोर्ड लाते हैं ना, जस्ट लाइक दैट होता है, उस पे एक लेयर पे वो होता है, कॉपर होता है।
कॉपर।
यस सर। और उस पे हम क्या करते हैं ना, जो हम सर्किट डिजाइन करते हैं ना सॉफ्टवेयर पे, उस सर्किट डिजाइन को उस पर प्रिंट कर देते हैं। प्रिंट करने के बाद एक फेरिस क्लोराइड का वो होता है, सॉल्यूशन होता है, उसमें हम इसको डिप कर देते हैं। तो जो हमने प्रिंट
क्लीन करना होता है।
या हाँ, तो जो रेस्ट ऑफ द पार्ट है जो मेरे जरूरत का नहीं है, तो उसको हम वहाँ पे क्या करते हैं, वो इरेज कर देते हैं।
Yeah, so I think this is the circuit जो आपने उस पे इम्प्लीमेंट किए हैं।
या यस सर,
सर्किट तो
यस
क्या
सर।
बता सकते हैं आपने क्यों बनाया ऐसा सर्किट?
ये सर्किट के मतलब कई पार्ट्स हैं, मतलब जैसे अगर मैं बात करूं दिस वन पार्ट की, तो ये पार्ट है इनपुट पार्ट। इनपुट पार्ट में क्या, हम इसको जो सर्किट है, इस सर्किट को हम फोन से कनेक्ट कर रहे हैं। और फोन क्या होता है, हमारा सेंसिटिव डिवाइस है, इसमें कुछ ज्यादा वोल्टेज नहीं दे सकते हैं, या अगर कुछ मतलब ज्यादा वोल्टेज चला जाए तो फोन खराब हो जाएगा मेरा। तो उसके लिए ये सर्किट जो है प्रोटेक्शन सर्किट है। इसमें हम सबसे पहले क्या है ना, जो डीसी सिग्नल्स होते हैं ना, डीसी सिग्नल इन द सेंस के क्या बोलेंगे हम, जैसे बैटरीज होती है ना, तो बैटरी की जो सप्लाई होती है वो डीसी सिग्नल होती है। और जो हम लोग नॉर्मल साइन वेव, अभी इस वीडियो जो प्ले हुआ होगा तो लास्ट में साइनोसोडल वेवफॉर्म आपने देखा होगा, ट्रायंगुलर वेवफॉर्म एंड साइनोसोडल वेवफॉर्म। ये सिग्नल सारे जो है ना क्या एनालॉग सिग्नल्स होते हैं, जो आपको जो है ना इसको हम देख सकते हैं। और डीसी सिग्नल्स हम इस फोन के अंदर सेंड नहीं कर सकते कि फोन खराब हो जाए, क्योंकि फोन की अपनी लिमिटेशंस है, क्योंकि फोन तो ऑसिलोस्कोप के लिए नहीं बना है ना, उसको नॉर्मल यूज के लिए बनाया गया था। हम उसको एनहांस कर रहे हैं कि हम उसको ऑसिलोस्कोप की तरह यूज कर रहे हैं, तो कुछ लिमिटेशंस फोन की है कि जैसे डीसी सिग्नल नहीं दे सकते। तो
हमारे घर पे जो लाइट आता है, जैसा
जी सर, यस सर,
लाइट, तो वो एसी होता है ना शायद?
यस सर, जी जी
AC
जी
होता है,
जी, यस।
तो वो अगर डीसी देंगे तो फिर वो सर्किट टूट जाएगा, राइट? क्योंकि
हाँ,
एसी और डीसी में क्या
बट
फर्क
सर,
है, कोई बता सकते हैं?
एक पॉइंट और मैं पहले बोल दूं, एसी सिग्नल भी इसकी लिमिटेशन है। जो मैंने जो सर्किट में कंपोनेंट्स यूज किए हैं ना, उसके कुछ लिमिटेशन है कि इस सर्किट में जो कंपोनेंट है उसको अगर उसके लिमिटेड वोल्टेज से अगर ज्यादा वोल्टेज देंगे तो वो खराब हो जाएंगे कंपोनेंट, है ना?
ओके।
तो उसकी लिमिट इस वीडियो में मैंने बताई थी कि उसकी लिमिट है कि हम 10 मिली वोल्ट से लेकर के 50 वोल्ट तक के सिग्नल को ही इसमें देख सकते हैं।
Okay, so this is the voltage range जो AC Jay was asking, तो जो AC सिग्नल है जो वेरी होता रहता है, तो वो 10 मिली वोल्ट से लेके 50 वोल्ट्स तक हम मेजर कर सकते हैं।
या यस सर,
तो आई थिंक कभी हम बीच में नर्व सिग्नल का भी वी वर डिस्कसिंग, तो नर्व सिग्नल का पोटेंशियल किसी ने कुछ बताया था, लाइक कैन एनीवन रिमाइंड मी हाउ मच इट इज?
minus 30 to 70 volt. minus 70 मिली नहीं मिली वोल्ट में, ओके
हाँ मिली वोल्ट -30 to -70 मिली वोल्ट्स, राइट?
तो आप वो भी मेजर कर सकते हैं क्या?
अभी इसमें नहीं कर सकते, इसको थोड़ा हमें अपग्रेड करना पड़ेगा। इसका लिमिट मैंने बताया ना अभी कि 10 मिली वोल्ट से 50 वोल्ट तक दे सकते हैं सिग्नल, नहीं हो पाएगा। तो उसके लिए थोड़ा हमें मॉडिफाई करना होगा, सिग्नल को और एम्पलीफाई करना होगा ताकि मैं अपने सिस्टम के से यूज कर सकूं, उसको सिस्टम के कैपेबल बना सकूं।
हाँ करेक्ट। एक्चुअली अगर आप देखेंगे लाइक इफ यू सी द एक्चुअल सिस्टम, तो दिस इज वेयर द इनपुट इज कमिंग। सो ये जो 2 पिंस है, so जो भी सिग्नल आप टेस्ट करना चाहते हो, सो आई थिंक अर्लियर Hina was asking us if you can test blood pressure or breath rate, whatever. So anything that you want to measure electrical potential, तो उसको इन दोनों के बीच में कनेक्ट करना रहेगा। Is that correct?
Yes sir. Yes sir.
Right.
और फरहान सर एक चीज और पूछना है कि कौन सा फोन हम इसमें यूज करते हैं, मतलब मिनिमल हमारी
सॉरी वॉइस ब्रेक हुई मैम आपकी। मैम यू आर म्यूटेड।
आप कौन सा फोन यूज कर सकते हो?
कौन सा फोन हम लोग यूज कर सकते हैं?
कोई भी स्मार्टफोन आप यूज कर सकते हैं मैम, क्योंकि मैंने एक एप्लीकेशन प्ले स्टोर से डाउनलोड किया, एंड्रॉइड के प्ले स्टोर से डाउनलोड किया मैंने।
कौन सा एप्लीकेशन?
और मैंने उसका नाम था वो ऑस्लोस्कोप करके एप्लीकेशन है। आप उसमें
क्या हाँ क्या ये ऑक्सीमीटर के जैसा है, सिमिलर है ऑक्सीमीटर के? ये क्वेश्चन था एक।
नहीं ये कह सकते हैं बस, क्योंकि बस ये सिग्नल को डिस्प्ले करता है और कुछ नहीं करता है। जो हमने जो इनपुट दिया ना बस आउटपुट देता है उसके सिमिलर, और कुछ नहीं करता है, इसका काम और कुछ नहीं है।
Okay.
So whatever signal you are giving, you can see it here on oximeter and blood pressure measurer, all those things. I think they have some additional equipment which sort of is suitable only for certain things, like
basically it's a very general purpose instrument है ये oscilloscope. I think right from instrument maintenance से लेकर के bio वगैरह I think हर application में काम आता है।
Ravi, one thing, this gives a graphical representation, right, on the screen.
Yes sir.
Of the current. So if there is any current which is there, it can be shown as a graph. So even ECG is a current, like in the heartbeat is a ECG. So
yes, it is used in ECG.
It's the same way of pattern, na? Like same pattern we get.
Yes, oscilloscope का वहाँ पे application होता है, like heartbeat measurement में.
So wherever electrical current is passing, we can use oscilloscope. Is it?
Yes sir, exactly.
एक और इसमें फरहान से आप बता सकते हो, फरहान ये नॉर्मली अगर आप मार्केट से खरीदने जाएंगे ऐसा इंस्ट्रूमेंट, तो क्या आपको कॉस्ट पता है क्या किस रेंज में आता है?
ये बहुत कॉस्टली इंस्ट्रूमेंट है सर। अगर कोई भी सबसे सस्ता भी खरीदने जाऊं तो 10,000 से ऊपर ही आएगा सर।
एग्जैक्टली, एंड ये आपने काफी इसको फ्रूगल वे में बनाया है। तो बेसिकली आई थिंक एक और आप इसको वैसे भी देख सकते हो ये प्रोजेक्ट में कि देयर आर सर्टेन लिमिटेशन, जैसे फरहान ने बताया कि 10 मिली वोल्ट से लेकर के 50 मिली वोल्ट तक, आई थिंक 50 वोल्ट तक इसका रेंज है। बट स्टिल मार्केट में जितने भी अवेलेबल है ना जो रेडीमेड इंस्ट्रूमेंट्स होते हैं, उनका कॉस्ट काफी ज्यादा होता है। एंड स्पेशली व्हेन यू गो टू कॉलेज एंड जब आप वहाँ पे किसी भी इंस्ट्रूमेंट के साथ में काम करते हो, वेदर यू आर अ बायोलॉजिस्ट और इवन एन इंजीनियर, अगर आप इंजीनियरिंग की पढ़ाई भी कर रहे हो, तो अगर आप खुद से अपना इंस्ट्रूमेंट डिजाइन कर पाओ भले विद सर्टेन लिमिटेशन, आई थिंक दैट इटसेल्फ इज अ वेरी गुड टेक होम पॉइंट, आई थिंक ये एफर्ट से।
But here see, I have one question. यहाँ पे आपको वोल्ट मीटर भी पता होगा ना? वोल्ट मीटर भी तो वोल्टेज को मेजर करता है। ऐसा ही होता है कि इसमें भी 2 इलेक्ट्रोड्स होते हैं और आपको स्क्रीन होती है, स्क्रीन पे आपको वोल्टेज मिल जाता है, राइट? वो वोल्ट मीटर होता है। तो इसमें और उसमें क्या फर्क है? मतलब ऑसिलोस्कोप और वोल्ट, क्योंकि वो भी वोल्ट ही मेजर कर रहा है और ये भी करंट को दिखा रहा है।
यस सर, बेसिकली फर्क इतना सा है कि अगर आप वोल्ट मीटर किसी जगह इस्तेमाल कर रहे हैं तो वोल्ट मीटर की जगह आप क्या करते हैं, एक सिर्फ आपको पर्टिकुलर वैल्यू मिलती है कि 4 वोल्ट है या 5 वोल्ट है या 10 वोल्ट है। बट इसमें क्या होता है ना, आपको फ्लक्चुएशन मिल जाएगा कि 4 वोल्ट 4.5 वोल्ट पर टिप हुआ था, फिर उसके बाद 4 वोल्ट आया,
या क्या, या फिर वो राइज कैसे हुआ सिग्नल, डायरेक्ट 5 वोल्ट पे आ गया या बीच में 3 वोल्ट पर रुका था, या कैसे हुआ था, सारी सिग्नल के टाइम टू टाइम कैसे सिग्नल वेरी, ये सारी चीजें हम देख सकते हैं। राइज टाइम, फॉल्ट टाइम क्या था हमारे सिग्नल का।
तो जय, अगर वही सिग्नल वोल्ट मीटर को हम देंगे ना, so what you will observe is voltmeter का needle will keep on moving. So जब high था वो एक साइड रहेगा, जब low था दूसरे साइड रहेगा, तो it keeps on moving, oscillating as a needle. Whereas what oscilloscope allows us is उसको एक waveform के form पे दिखाएगा. So this also sort of measures, but you cannot use it in any way. जो
आप मतलब उस वैल्यू को मतलब आप नोट नहीं कर पाओगे अगर मल्टीमीटर से कर रहे हो, मगर ऑसिलोस्कोप से करेंगे तो नोट कर सकते हैं उस वैल्यू को।
You can
Yeah, so pattern also you will know. Fluctuation का pattern क्या है?
Yes.
Yeah, yes sir.
And I think many of the signals, biological signals, for example, EEG जैसा या EMG जैसा, like fundamentally principle is very similar. Like you have some electrodes, ये probes करके जो बनाए हैं, so you'll insert those electrodes and you basically have some mechanism by which आपको वो signals दिखेंगे. Like you would have seen film में या real में भी आप देखे होंगे, like कुछ monitors रहे होंगे electrical hospital के machine के बाजू में, like so it's a very crude analogy, but analogy like similar instrument है, but it requires lot more electronic components to be able to measure minor changes, and that's what I think जो इसका limitation में Farhan was mentioning, इसका specifications वगैरह और refine कर सकते हैं for different applications.
So here the only circuit will be आप अगर किसी वोल्ट मीटर का सर्किट लेके इसके सर्किट से कंपेयर करोगे तो थोड़ा ही चेंजेस होगा ना, क्योंकि दोनों का काम एक ही है।
थोड़ा नहीं, ज्यादा ही
डिफरेंस है, क्योंकि हम यहाँ बस क्या कर रहे हैं, सिग्नल को ले रहे हैं और सिग्नल को फोन तक पास कर रहे हैं। जितने प्रोसेसिंग के प्रोसेस हो रहे हैं ना, ए टू डी कन्वर्जन, या मतलब क्योंकि सिग्नल बेसिकली एनालॉजी में क्या है, एनालॉग है, बट जो हमारा फोन है, फोन पे तो एनालॉग सिग्नल तो वो दिखा नहीं पाएगा, वो सारे सिग्नल क्या होगा, कन्वर्ट हो रहे हैं डिजिटल में, फिर वो एनालॉग में वो सैम्पलिंग हो रही है, फिर आउटपुट दे रहा है आपको। तो इसके सर्किट और उसके सर्किट में फर्क होगा थोड़ा।
Yes, so there is going to be a lot of additional complications or sophistication needed. Just as an example, like we can think of sensation and action happening at the level of microorganism also. Like एक paramecium या bacteria है, it senses and it acts. But उसमें जो mechanism है, and say in a snail or in a mammal, तो दो description level पे you can think of the same mechanism. There is sensation happening, there is some response, right? So but there is sophistication or the sensitivities are completely different. So ये देख नहीं पाएगा, it can just sense some chemical gradient or whatever, whereas higher organisms have different senses, right? So the probes will have some sophistication attached. So but it is a good model to think about all equipment, all ranging from EEG and various other things and various applications that he showed us about.
Yeah, correct. And you can also like make a model like how easily we can learn to make circuits, like you know, because see this circuit looks very complicated to everyone. मुझे भी बहुत complicated लग रहा है, सब लोगों को शायद देख के लगता होगा कि ये क्या है ना, अलग अलग ये क्या है। तो आप कुछ अलग से हम डिस्कस कर सकते हैं ग्रुप में।
यस सर,
और हम
बहुत इजी इसको बनाना सर।
अलग अलग सर्किट कैसे हम लोग अच्छे मतलब सिंपली हम लोग सर्किट को सीख सकते हैं, मतलब एक दो मॉडल को देख करके फिर उसमें से सर्किट इजीली भी बना।
यस प्रशांत का एक लास्ट क्वेश्चन ले सकते जय, आई थिंक उसके बाद वेदर स्टेशन का भी एक है।
यस मोबाइल पे कैसे कनेक्ट करेंगे, लेकिन यूएसबी माइक्रो यूएसबी से कैसे कनेक्ट करेंगे?
सॉरी मैंने वीडियो में एक स्लाइड रखी थी ऑक्स केबल की, 3.5 mm जैक था, ऑडियो जैक। दिस वन आपने एयरफोन में यूज किया होता है, एयरफोन में देखा होगा अपने फोन के एयरफोन में, है ना?
यस।
तो इसके थ्रू हम कनेक्शन करते हैं फोन में, क्योंकि हम जो एयरफोन में क्या होता है, माइक होता है ना, माइक,
हाँ,
तो माइक
इसको
क्या है
हम
आपके,
दो डायोड कनेक्ट करते हैं क्या?
सॉरी,
इसको डायोड जो प्रोब है वो कनेक्ट करते हैं क्या, कैसे कनेक्ट करते हैं?
वही
तीन
वही
केबल
बता रहा
रहते हैं
हूँ,
ना।
वही बता रहा हूँ। तो इसमें जो 4 रिंग है ना, 4 टिप, रिंग एंड स्लेव, तो क्या होता है इसके जो अगर मैं नॉर्मल वे में बताऊं एयरफोन के तरीके से, तो क्या होता है, पहली वाली जो टिप होती है ना, दिस वन, फर्स्ट वन, तो ये क्या होती है, ऑडियो के लिए होती है, राइट या लेफ्ट ऑडियो के लिए होती है। फिर दूसरी वाली दूसरे ऑडियो की, एक राइट के लिए एक लेफ्ट के लिए, ठीक। उसके बाद जो थर्ड वन होती है वो अगर मतलब थर्ड वन है तो उसमें वो होता है माइक के लिए, और फोर्थ वन होती है ग्राउंड के लिए। ये एयरफोन के जैक का कनेक्शन होता है। तो मैंने क्या यूज किया, मैंने सिर्फ माइक और ग्राउंड का कनेक्शन यूज किया, बाकी कनेक्शन वैसे ही छोड़ दिए नॉर्मल, क्योंकि वो आउटपुट टर्मिनल से, इनपुट टर्मिनल नहीं है। माइक का क्या है, माइक हम जब कुछ बोलते हैं तो माइक के थ्रू क्या जाता है, फोन में जाती है, कोई डेटा जाता है ना। मोबाइल को कोई डेटा सेंड करनी है, हमको ऑडियो भेजना है, तो कैसे जाता है, माइक के थ्रू जाता है। तो इसलिए हमने माइक और ग्राउंड का पिन यूज किया है सिग्नल को सेंड करने के लिए।
Okay. So what he's basically saying is, because we want to send some signal into the mobile, if you look at mobile, sound is an output for a mobile or for any computer or any device, whereas mic is an input device, right? अभी आप लोग भी जब यूज कर रहे हैं, whatever you are hearing is output from the speaker, and whatever you are saying is input to the computer, to the mobile. So he wants to give something into the computer or mobile. So he is using mic and ground as two things जिससे वो मोबाइल को इनपुट दे सकता है। प्रशांत, जो भी आप लाइंस बनाए, कैन यू प्लीज रिमूव देम बाय undoing? आप डिलीट बटन जो है वो प्रेस कर सकते हो।
मैंने नहीं बनाया।
Okay, no issues.
So if there is so much of noise, or I want to record larger signals, so how I can record it?
हेलो,
हाँ फरहान, लार्जर सिग्नल्स अगर सेंस करना है, हाउ कैन यू रिकॉर्ड इट?
लार्जर सिग्नल इन द सेंस,
Lokesh, you can please repeat.
हाई फ्रीक्वेंसी का।
हाई फ्रीक्वेंसी रेंज भी मैंने, सर कैन यू प्लीज चेंज द स्लाइड सर?
या
second last slide,
सेकंड लास्ट स्लाइड,
just before the application.
मैंने यहाँ फ्रीक्वेंसी रेंज लिमिट बताया है, क्योंकि फोन के जो लिमिटेशंस है, क्योंकि वो मैंने तो ऑसिलोस्कोप बनाया नहीं ना फोन, कि फोन पहले बनाया है, तो उसके जो लिमिटेशन है उसके अकॉर्डिंग 75 हर्ट्ज टू 15 किलो हर्ट्ज तक का सिग्नल को ही हम देख सकते हैं।
Okay. So 75 hertz is the lowest frequency and 15 kilohertz is the highest frequency of
yes, sir.
the signal. So जो भी सिग्नल इसको हम इनपुट दे रहे हैं, तो वो उसका फ्रीक्वेंसी, दैट इज नंबर ऑफ सिग्नल,
इससे कम की फ्रीक्वेंसी भी देख सकते हैं, इससे ज्यादा की फ्रीक्वेंसी भी देख सकते हैं, बट हम उसको कैलकुलेट नहीं कर पाएंगे, क्योंकि उसमें इस तरह से सिग्नल नॉइजी टाइप कह सकते हैं उसको। ये जो विजिबल है, मतलब हम इसको कैलकुलेट कर सकते हैं बेसिकली सिग्नल।
So this is where I think the measurement will be reliable. उसके आगे और पीछे it's going to have some the signals will become poor. So you can still see, but you cannot.
आप इससे वॉइस का फ्रीक्वेंसी भी मेजर कर सकते हैं।
या श्योर सर,
बर्ड का कॉल का फ्रीक्वेंसी
बिल्कुल आसानी से कर सकते हैं, कोई दिक्कत नहीं है।
बर्ड का I think is a very good example.
lot of audios of bird recording, bird call recording, right?
Yes.
तो आप फ्रीक्वेंसी से बता सकते हैं कौन सा बर्ड है ये?
ये मुझे प्री डिफाइन करना होगा कि फ्रीक्वेंसी मतलब किसकी क्या फ्रीक्वेंसी है, उसके अकॉर्डिंग बता सकते हैं।
तो बर्ड के वॉइस से हम लोग बर्ड को आइडेंटिफाई कर सकते हैं ना? या बट एक ही बर्ड के डिफरेंट वॉइसेस को पता कर सकते हैं, उसके बिहेवियर को इस तरीके से मार्क कर सकते हैं।
हाँ, क्योंकि हम
अगर हम लोग को पता हो कि फ्रीक्वेंसी कितनी है, तो वी कैन डू दैट, आई फील, विद दिस
Okay, so should we move to weather station?
Yes. Let's we can move. Yeah.
Yes. So Kiran, can you please?
Yes, yes.
So next
Yes,
is
I can.
weather station is the next thing which to sense. Now actually we have sensed the electrical signal. We will use for weather station for measuring the weather, sensing the weather.
Various parameters. Yes.
Various parameters.
Hi everyone.
Hi Farhan. Please play. So what are you going to tell us today?
My project, we make a project in IoT based weather station.
Okay. So from the name, it sounds like you have made some sensors and various other things to measure parameters in the weather. So what all things does this sense?
It's a combination of sensors. We are measuring some environmental variables included atmospheric pressure, temperature, humidity, rainfall, wind speed, light intensity, etc.
Wow, so it's quite a lot of things. So can you show us what all is there moving ahead, Farhan? What is it?
Yes, sir. One second. This is a complete view of my project.
Okay, so can you show us where each sensor is? So what is this one?
This one is has temperature and humidity sensors.
Okay.
DST11 and BMP280 sensor.
Okay, temperature and humidity is sensed by this. It looks cool. And?
Yes, sir. Sensors names are BMP280.
Okay.
And DST11.
And DHT11. Yeah.
DST11 is a temperature and humidity sensor, and BMP280 is a atmospheric pressure and temperature sensor.
Okay, so there are two temperature sensors. Cool.
Yeah, yes, sir.
What else? You said some rainfall, wind speed, various light intensity, various.
Yeah, yes, sir. In this one is blue color box is a rain gauge. It measures the rainfall.
Okay.
And this one is wind directions.
Okay, wind vane.
Yes, sir. This one is wind speed anemometer.
Okay.
To measure wind speed. This box it has a microcontroller and light intensity sensors.
Okay. So let's move ahead and show us how you made this, and then we will quickly see where this will be applicable for a lot of projects that our friends are also working on.
Yes, sir. Please change slide.
Yes.
Yeah. This slide is how I make a project.
Okay.
This is a circuit diagram and circuit diagram 3D view. How actually PCB look like.
Okay, so I think this is something that all of us would be familiar with when we open some electronic things. You see some green board. So as Farhan is saying, so it's a very this is the way how we implement a circuit, so which has a lot of components. Sir. And let's move ahead, Farhan.
Yes, sir.
Okay, so
this is
live. Yes.
Yes, sir. Physical structure how actually look like a circuit.
Okay.
This is the various part. The first one is this one is rain gauge. This one is for using temperature and humidity sensor and atmospheric pressure sensor. This is an anemometer.
Okay.
This one is an internal mechanism of rainfall. Rain gauge, how rainfall. This one is wind speed measurement.
Okay.
This is some graph of wind speed and rainfall.
So how do we see these things as a graph, Farhan? Like we have a lot of sensors here, so I'm assuming it has some electronic components, some mechanical components, which sort of captured it, and how do we see them as graphs finally here? So can you quickly tell us something about it?
Yes, sir. This graph is plotted by server. I am using a ESP32 to sending a data. I am using ESP32 sending a data to server.
Okay.
Server. I use a server name. Server name Things board.
Okay. So you are sending all the data that is collected by the microcontroller. I'm guessing ESP32 is a microcontroller,
Yes,
right?
yes, sir, with Wi-Fi module.
Oh yes. So we don't need to have wires and others. So this component is capable of sending the data via Wi-Fi to a remote server. So
Yeah,
I
yes,
think
sir.
there will be more questions on it which we will discuss after this video, but let's move ahead. So we will see those things sent here in the server, and we have the we have the data as graphs on the server. Yes.
Yeah, yes, sir.
So what more? So we see rainfall and wind speed plotted here, and we see temperature here. What more do we
Yes,
have?
sir. One is BMP temperature and one is DST11 temperature graph.
Okay. So can you show us, do we have plots of other things as well here?
Yeah, yes, sir. Once again. This is the wind speed meter.
Okay.
And second one is the humidity.
Okay, so wind speed is in meters per second. It's about 2 meters per second, and humidity we have at
percentage.
54%.
Yes, sir.
And
The second one is light intensity.
how
light
bright the day
intensity.
is. Okay.
Yes, sir.
Yeah. And
This is
pressure and putting things together again, I think.
Yes, sir. Yes, sir. All the graph combined together.
Yeah. So,
First one,
yes, Farhan, please.
first one is a humidity level graph, and second one is light intensity graph.
Okay. So, are there any more graphs?
No, sir.
Okay. So as we can see here, I think what Farhan along with his friend Firoz has shown. So there are quite a lot of parameters that they are measuring, and there is parameter 1, parameter 2, parameter 3, parameter 4, parameter 5. So how many? 5, 6? How many, Firoz or Farhan?
Approximate 6 approximate.
6, yeah. So we have 6 weather parameters, and all of them are sending data to a microcontroller. What is its name?
ESP32.
Yes, it's ESP32, and from here it's sent out through Wi-Fi to a remote server, and from the server you can access them on your any device and see those parameters remotely. So I think this is what is dominantly understood as Internet of Things. That's why Farhan told us IoT design in the beginning. So you can have sensors all across and collect information in a server and see them here. So tell us, Farhan, like where all do you think it's going to be useful?
So first, it is used in agriculture applications. Maybe it may be installed on a field of farmer.
Yes.
So he can monitor its field temperature and humidity. So for irrigation system it may be helpful.
Yeah, weather is very important for agriculturists. I think they need to know
Yes,
when
sir.
it's going to rain and stuff. Yes,
And maybe it may be installed in a office or company. When you go to the company and whether you know the weather of the company is how it's changing, and if it's raining, you prepare for the rain from home.
Yeah, so I think it's useful. I think in any workspaces, offices and companies and others, as Firoz is saying,
Yes, sir.
where you can sense various weather parameters and inform the people. So I interestingly see laboratory as well here. So what exactly do you have in your mind, Farhan and Firoz, when you say laboratory?
So basically laboratory is इसलिए use किया because we are using when we experiment something we need a मतलब fixed parameters for temperature and humidity, that thing.
Okay.
So that's place we are also use.
Okay, so basically like in our experiments and others, I think a lot of cubists here would be aware they need to keep track of temperature, humidity and various other weather parameters. And later today when we are going to see the mango mapping and others, like where seasonal variations have lot of effect on trees and how various things in our environment behave. So we need to have a close eye on how the temperature, humidity, pressure, rainfall, so all these parameters. And I think in those applications as well, things of this kind could be very useful. So I think we will have more questions from these things. Thank you, Farhan, Firoz, and I think Ashish also has helped the students in making this. So let us have an interesting conversation around this moving ahead. Thank you.
Yeah, thank you, sir.
So any questions? Yeah, we can take questions.
Hello.
Please
I want
ask
to
questions.
ask that what is the cost of IoT?
Sorry.
I am
I didn't
asking
get
that
point.
cost of IoT.
I didn't
IoT
get point. IoT cost.
No,
Module.
Farhan, he's trying to say to what is IoT means.
नहीं, ये पूरा सिस्टम बनाने के लिए आपको कितना कॉस्ट लगा ऐसे? आई थिंक सो इज आस्किंग.
मतलब IoT weather station cost. Okay. 1,500 rupees approx. Hello. Hello.
किट IoT का किट 1500 में मिला क्या?
नहीं, मैंने मतलब बनाया है, उसमें आधी चीजें मैंने खुद बनाई है बेसिकली.
फिर कनेक्ट कैसे किया थिंग्स को, इसको थिंग्स पैड को?
थिंग्स बोर्ड एक सर्वर है, मतलब सर्वर इन द सेंस क्या है कि एक मतलब हम जैसे कोई चीज सर्च करते हैं ना गूगल पे, वैसे एक सर्वर है थिंग्स बोर्ड.
तो उस सर्वर we can freely upload there. Upload our data on server. We can freely upload our data on server there.
या कह सकते हैं.
Hello. So how
Yeah.
we can use this IoT for agriculture? Means specific. बताओ थोड़ा.
सर जैसे बेसिकली फील्ड में सर इरिगेशन सिस्टम जब चलता है, मतलब सर पानी के लिए जब पानी है तो सर उसके लिए सर ह्यूमिडिटी कितना है, टेम्परेचर कितना है, मॉइस्चर ऑफ एयर कितना है, उस पे इफेक्ट पड़ता है सर फील्ड के ऊपर, क्रॉप्स के ऊपर. तो हम इससे सर इरिगेशन उसके सिस्टम पर
मतलब ये भी देख सकते हैं कि कितने दिन पहले बारिश हुई थी, कितना बारिश हुई थी, ये सारी चीजें, फिर हमें पानी देना चाहिए कि नहीं देना चाहिए, ये सारी चीजें हम इसके थ्रू यूज कर सकते हैं. इसमें वहां ऐसे यूज हो सकता है.
हाँ, तो फिर जो वाईफाई सिग्नल जाता है तो हमें कंटिन्यूअस इंटरनेट चाहिए क्या वो सिग्नल डाटा सर्वर के लिए चाहिए देने के लिए?
यस सर, मुझे वाईफाई चाहिए होगा. नहीं तो हम वहाँ पर GSM मॉड्यूल भी लगा सकते हैं अगर वाईफाई वहाँ पे नहीं है.
सिम कार्ड यूज कर सकते.
या
सिम कार्ड यूज कर सकते. और पूरा टोटल फिर एग्रीकल्चर के लिए बनाने के लिए हमें 1500 में हो जाएगा ना वो?
या
मॉड्यूल का सिर्फ ना, बाकी का हमें खुद बनाना पड़ेगा ना वो?
नहीं पूरा कंप्लीट बना उना के कंप्लीट मैंने जब कंप्लीट कर लिया उस तब की बता रहा हूँ पूरा.
वो व्यू वाला सब, म्हणजे व्यू वाला, वेदर का सब, टेम्परेचर का
या पूरा कंप्लीट बॉक्स पूरा कंप्लीट करके.
हेलो फरहान, क्या आप इससे ह्यूमिडिटी भी मेजर कर सकते हो?
बिल्कुल मैम,
या
बिल्कुल।
फिर कुछ अलग पैरामीटर्स या कुछ अलग सेंसर्स यूज करना पड़ता है वो एक मेजर करने के लिए?
हमने DHT11 सेंसर यूज किया है जो उसमें ह्यूमिडिटी एज वेल एज टेम्परेचर दोनों मेजर करता है।
हाँ, बट तो कैसे मेजर होता है इन द सेंस कि आपको ह्यूमिडिटी और टेम्परेचर के लिए व्हाट डू यू हैव टू मेजर, लाइक व्हाट पैरामीटर यू विल मेजर फॉर ह्यूमिडिटी, व्हाट इज द प्रिंसिपल ऑफ वर्किंग?
क्या ये इंस्ट्रूमेंट के पीछे कोई प्रिंसिपल है जिसके ऊपर ये प्रिंसिपल के ऊपर जो ये इंस्ट्रूमेंट वर्क करता रहेगा, ऐसा कुछ है?
वो सेंसर फरहान यू कंटिन्यू।
नहीं, कंटिन्यू बोलो फिर।
बेसिकली हमने सेंसर यूज किया है जो सीधा सेंस करके हमको डिजिटल सिग्नल के रूप में उसको हम दे देता है हमको, तो उसकी वजह से हम
बरोबर है, बट आई एम आस्किंग कि व्हाट इन टेम्परेचर इट मेजर, वेदर इट मेजर्स द हीट, सो व्हाट इन हीट इट मेजर्स, लाइक
Farhan and Feroz, like the question जो यहाँ पे पूछ रहे हैं वो ये है कि DHT11 सेंसर के अंदर टेम्परेचर और ह्यूमिडिटी को मेजरमेंट करने के लिए क्या यूज हो रहा है, वो हमें किस टाइप का डेटा दे रहा है? Is it like your change in voltage with respect to temperature, और क्या ह्यूमिडिटी के लिए कुछ कैपेसिटेंस चेंज हो रहा है?
यस, वोल्टेज चेंज हो रहा है ना, जैसे हम वो थर्मिस्टर, जस्ट लाइक थर्मिस्टर के बारे में कह सकते हैं, जैसे थर्मिस्टर हम यूज करते हैं टेम्परेचर के लिए, तो उसमें क्या होता है, रेजिस्टेंस चेंज होता है, जैसे ही इसमें भी वो वोल्टेज चेंज हो रहा है।
बेसिकली व्हाट इज हैपनिंग जय इज जो भी टेम्परेचर चेंज हो रहा है या ह्यूमिडिटी चेंज हो रहा है, वी नीड सम मटेरियल जो उसको एक इलेक्ट्रिकल चेंज में कन्वर्ट कर पाए।
व्हाट इज एक्चुअली इट चेंजिंग लाइक दैट?
अभी जो फरहान बोला है थर्मिस्टर, आई थिंक कैन यू एक्सप्लेन इधर कैसे चेंज होता है फरहान?
मतलब थर्मिस्टर जो बना होता है ना सर, वो ऐसे मटेरियल का बना होता है जिसमें क्या होता है कि जैसे जैसे उसके हीट उसके पास लाते हैं थर्मिस्टर मटेरियल के पास, तो उसका रेजिस्टेंस कम होता चला जाता है। रेजिस्टेंस जब कम होता है तो उसके अकॉर्डिंग हम उस पर कैलकुलेट करते हैं कि टेम्परेचर राइज होने पे कितना रेजिस्टेंस घट रहा है या बढ़ रहा है, उसके अकॉर्डिंग हम उसको बता पाते हैं कि इतना टेम्परेचर है या इतना मतलब ह्यूमिडिटी है, जस्ट लाइक दैट।
Yeah.
So its physical property should change, right? That way.
Yeah.
So
what about humidity then? Then Achal was asking about, I think, humidity.
Yes. Humidity का कैसे होता है फरहान?
Humidity का actually I don't have an idea, sir.
किसी के पास कुछ guess है?
Farhan, think in terms of capacitance. So there is a
yes,
capacitive sensor for humidity inside the DHT. So जिस तरह like your
मतलब मुझे वो मोटा मोटी पता, मतलब एग्जैक्ट मैं वो मेथड मुझे नहीं पता, मतलब तो गलत बताने से अच्छा है कि बोले आदमी था।
नहीं चलेगा, बताओ। यहाँ गलत सही कुछ नहीं है, we'll just think logically and try to figure out कि
गलत बताओ, लोग कंफ्यूज हो जाएंगे। अच्छा होगा।
What is humidity? What is humidity? Humidity क्या होता है? Audience, we'll just discuss this and I think we can close.
So, if we talk about humidity, it's like we were having a discussion on climate. Sorry for the background noise. We were discussing about climate, whether climate really has a
Yeah, but what is humidity in the climate?
So,
moisture
content.
Moisture content. Yeah, so humidity is basically moisture, right? So we'll have to, I mean, one intelligent guess that we can make is there is something inside this which changes its physical property, as Jay said, when it becomes wet, when there is moisture, and it changes it very sensitively, right?
Yeah, sir.
Yeah, so yes, Firoz, you want to say? Yeah.
तो जब उसमें सब जैसे वो ह्यूमिडिटी ऑब्जर्व करता है सर, जैसे मॉइस्चर ऑब्जर्व करा उसमें, तो सर उसका कैपेसिटेंस इंक्रीज हो जाएगा। जैसे सर अगर कोई भी मटेरियल अगर कॉपर है सर, वो जैसे ही रस्ट होता है तो उसका रेजिस्टेंस इंक्रीज हो जाता है। ऐसे ही सर इसमें सेंसर के अंदर इनबिल्ड ऐसा मटेरियल है जो कि उसको एब्जॉर्ब करके सर उसका कैपेसिटेंस वैल्यू चेंज कर रहा है और हमको आउटपुट में दे रहा है।
ओके, सो कैपेसिटेंस जैसा just the way the resistance changes with heat in a thermistor. So capacitor is basically two plates in between which there is some material, जिसको हम dielectric material बोलते हैं. But
DP, I'm not from electrical. Well, I don't from electrical background. I'm using my 12th what I've studied. So is it like increasing of the dielectric constant and thereby increasing the capacitance of
yes. So the material inside, so depending upon how wet it becomes, so uska, how easily can it pass electric field between the plates, it changes. So there is some complex physics behind it, which we can discuss sometime when you all join. But that's basically what is happening here, as Bivas just said. So dielectric constant of this material changes with the moisture, and in turn the capacitance of this entire setup changes, and which gets reflected as a change in electric signal, जो यहाँ से निकल के हमें sense. So the same is the principle everywhere. Like whatever you are measuring, whether it's oximeter or whatever you are measuring, so some physical change needs to be converted into electrical change, and different material can be used in different places. Yeah.
So basically the dielectric over here is some material which is actually like absorbing your moisture. So the more moisture and so it is the middle layer, and the outer two layer are you can consider it as electrodes.
Yeah.
So so they're simply passing some of the charges right from one end to another. So
And an interesting connection here is this is the same thing that's happening with all our sense organs.
Yes.
Because as we have seen with snails and others, and we discussed about nerve signals and others very briefly, but all our sense organs basically, like light aa raha hai, so there is something in our sense organs which convert it into electrical pulse or create some potential.
It's ionic change. Here there is a chemical signal. So
yes,
ionic change, a property of chemical property is changing. Same is for pH meter also. Like for pH meter there is a hydrogen ion concentration is what matters. So the flow of charges change, and then the chemical property changes, and then the it's actually showing as electrical change.
Basically like converting your one physical form of energy to the electrical energy.
So this is basically an organism which has 6 sense organs, and those sense organs are which senses heat, moisture, which is humidity, rainfall, speed of the wind, direction of the wind. So it's basically an organism, and a lot of processing is happening at the center using Arduino kind of a thing. So the thing is, like, it doesn't have any motor things right now, but we can always have, just the way Firoz showed us in the snail case. So movement can also be brought in, but very crudely, I think that's what we can understand. Yes, and this eats electric power instead of carbohydrates and dal chawal, idli, sambar. Yeah.
And that electrical power we can also derive from the sun energy.
Yes.
The solar energy.
Yes, because solar is also, I think, solar panel. If you give an example, solar panel, maybe there's some kind of that only. No? The heat is outside is actually thermistor. I think I don't know.
Photovoltaic cells.
Photovoltaic cells. Okay.
Yes. So this
works on the principle of same kind of.
Yeah. So this is what is referred to as transduction, as Gian is also mentioning. So it's the same principle. Yeah. So it's the same principle, whether it's your eye, ears, skin, or whatever, proprioception and various other senses that we have. So it's pretty similar.
Yeah. And like we can also use the term transducers. So these are the transducers.
For sensors. Yes. Okay. So should we then pause and move to the next system?
Yes, yes, we can move. So, which is next?
Over to
Is it
Jay.
fruit fly or?
Thank you, Farhan and Firoz. I think it's very good, and I
Okay,
think
sorry, I want to do something interesting here because we have discussed already about weather. Hello? Weather, temperature, and light, photon, and all solar and all. So I just want to bring mango before. If is it possible if I can bring, because it is connected to weather station. So because we have a weather station thing, and in which we have discussed about weather, and mango is also connected with the weather.
yeah, we could do that. That's actually the schedule now. But we were thinking that at the end there can be some activity where whoever is interested can continue with us, and so that's what we were thinking. But so you can take a call and let us know, like fruit flies or mango.
Yeah, they can do. You can tell them.
If the group is there already, then we can take. I don't think the group is here.
Mango group is not there.
Yeah, so we can take a fruit fly, and after that we can take up.
Okay, fine. Then we will have a fruit fly. Group is not. So let's have the fruit fly presentation next, because so it is. They are going to talk about. I think the research question is circadian rhythm, which is again a Nobel Prize winning work. All of the previous ones are also Nobel Prize winning organisms, and fruit fly is one of the major Nobel Prize winning organism. So over to fruit fly group. Is the presentation ready? They can present it. Show it. Hello.
Yes, Garima. Yes, please take over.
It's coming.
We have little scarcity of time, so let's little bit.
Right.
Let's start.
So, fruit fly group, you want to introduce something?
Hello. Yeah, just quickly, as Jai sir was saying that fruit fly is one of the popular model systems, I would just like to say we in the lockdown period, all cubists from different parts of the country have tried to work with this model system and address not only circadian rhythm, but do some jugards with the fruit fly model system, and you'll surely see it. Yes. Yeah, you can play the video. Thank you.
What is this, Rachel? Why are you wasting our time when we are supposed to do a lot of research work?
Oh, come on, man. You never understood me. What do you think I am doing?
What I can see is that you are inviting thousands of flies around us.
You? How dare you call them just flies? Don't you know what they are capable of?
What?
Then see. And you, my friends, you are also invited to see.
So why fruit flies? Fruit flies can be found easily on rotten fruits and vegetables. Here is an example. You can see fruit flies on mango, jackfruit and tomato. Fruit flies here are also seen on banana, onion and ginger. So they are easily available. Fruit flies have a short life cycle. The life cycle starts with adult male and female, which mate. The female lays eggs. The eggs hatch into larvae, and the larvae grows and feeds. The last stage of the larvae then develops and forms the pupae. And from the pupae, the progeny flies arise. Not only that, but large number of progenies from single mother can be obtained. Last but not the least, they're easy to maintain at homes. We'll see how. But before that, it's time to celebrate the 6-time Nobel Prize winner. Since 1900s, fruit flies have been used for research and they have been benefited mankind. Not only that, in 1900s, T. Morgan was awarded a Nobel Prize for his discoveries concerning the role of chromosomes in heredity. His student, H. Mueller, was awarded a Nobel Prize in 1946. Mueller was awarded Nobel Prize for discovery of the production of mutations by means of X-ray irradiation, a new technique for developing mutants. The third Nobel Prize was awarded to Lewis in 1995 along with Christine Walhard and Eric for the discoveries concerning the genetic control of early embryonic development, which means how eggs develop and what are the genes responsible for developing. The fourth Nobel Prize was awarded to R. Axel along with Linda in 2004. They were awarded the Nobel Prize for the discoveries of odorant receptors and organization of the olfactory system. The scientists deciphered the olfactory system and the odorant receptors of fruit flies and also human beings. The fifth Nobel Prize was awarded in 2011 to J. A. Hoffman. Hoffman was awarded the Nobel Prize for the discoveries concerning the activation of innate immunity, a current hot topic. Lastly but not the least, the 2017 Nobel Prize was awarded to J. Hall, Michael Rauschenbach and Michael Young for the discoveries of molecular mechanisms controlling the circadian rhythm.
Our foremost and immediate objective is trapping Drosophila melanogaster from our locality. But why Drosophila melanogaster? It is one of the fruit fly species that is being studied all over the world. It is being used in research for a very long time and its genes are 75% similar to that of human beings. Starting with trapping of fruit flies.
How to trap fruit flies? For trapping fruit flies, we require plastic bottle, any fruit or vegetable, and a cloth or a tissue paper. First, take fruit or vegetable. Here, banana is taken. Smash it or chop it into small pieces. Then, take a clean and dry plastic bottle and add the smashed or chopped fruit into it. Now, take a cloth or a tissue paper and clean the mouth of the bottle so that flies will not stick to the mouth and enter inside the trap. Now, keep the bottles near dustbin or kitchen areas. Observe the bottles after 1 or 2 hours.
Earlier, we used to prepare standard cornmeal agar medium for culturing fruit flies, but now, due to lockdown, the ingredients required for making the standard cornmeal agar medium are not available at our homes. So, for culturing and mentoring fruit flies at our homes, we all collaboratively design a new homemade medium called the tomato rava sugar vinegar medium as an alternative for standard cornmeal agar medium.
Homemade media for fruit flies. We will need 1 egg-sized tomato, 1 tablespoon sugar, 2 tablespoon rava, 150 ml water and 15 ml vinegar. Take 1 egg-sized tomato and chop it into small pieces. Smash it into a paste like this. Pour 150 ml of water in a vessel and keep it on a low flame. Add 2 tablespoons of rava and mix it continuously. Add 1 tablespoon of sugar. Now add tomato paste and cook the medium for 10 minutes on a low flame. After cooking, take the medium off the burner and let it cool for about 5 minutes. After 5 minutes, add 15 ml of vinegar and mix it in the medium. Transfer medium in clean bottles using a spoon. Allow the media to cool and solidify. Our homemade TRSV media is ready. These are a few samples of TRSV medium bottles prepared by different cubists.
Moving forward with identification and cube photomicroscopy. Fruit flies are identified by classifying them under different categories like kingdom Animalia, phylum Arthropoda, class Insecta, order Diptera, family Drosophilidae, further to genus and species identification. But is microscopic identification possible at home? Yes. In this lockdown, we developed a method of mobile photomicroscopy. And by looking at these images, we could say how well we could develop the mobile photomicroscopy.
Immobilization. Collect flies from single line culture. Put into freezer for immobilization. After 1 hour later, took in the bottles from freezer. Observe most of the flies were died in the bottle. Taken flies for identification purpose.
Requirements for a mobile photomicroscopy. A mobile phone, white sheet paper, transparent sheet paper, needle, a torchlight, and an immobilized fruit fly. Starting from the bottom, a torch, white paper, transparent sheet, object which is the wing of a fruit fly, and a phone camera. This is how the setup looks. And here is the picture of a wing from mobile photomicroscopy.
Now, moving on to the activity pattern, the circadian rhythm of fruit flies. Do you want to know when I wake and when I sleep? Here's my buddy with an experiment. So for this experiment, a tomato piece was used as a bait to attract the fruit flies. The aim of the experiment was to observe the number of fruit flies that have been attracted throughout the day. The observation was made for 2 days, and we could see that there was both increase and decrease in the number of fruit flies throughout the day. We could also observe the fruit flies moving around the bait, and also the activities were recorded. We do know that, like humans, the biological clock of fruit flies are triggered by the expression of circadian genes. The activities observed throughout the day are feeding. The fruit flies feed on the bait. Courtship behavior. Mating. Female and male fruit flies mate. Egg laying. The female fruit fly lays egg on the bait that they feed on. Defecating. With these observations made, a graph is plotted wherein the x-axis denotes the time lapse of 48 hours. The y-axis denotes the number of fruit flies observed. The pattern seen in the graph is followed by fruit flies and is same on both the days. The same patterns are being followed by the fruit flies and is observed by us for 2 weeks.
It is said that never repeat the same mistake again. But for that, you need to remember your mistakes. So here are some of the goof-ups that we made. There was a contamination in the media because we did not add the antifungal agent vinegar. We mistook an adult female fly to be gravid. A male fly was taken for single line cultures. Measurement of ingredients was not proper, leading to excess water in media and making it liquidy. In mobile microscopy, light source was inadequate and wrong direction for image capture. We mistook a phorid fly to be a fruit fly, which is a completely different family. Fruit fly has 6 Nobel Prizes to its name. It serves as a brilliant model for many different fields of study. One of them is neurobiology. Mutant flies show symptoms for neurodegenerative diseases like Alzheimer's and Parkinson's. We can also study evolution by doing the olfactory assay, comparing between lab-bred and wild flies. We studied how to identify these flies based on their wing morphology. Working from home, we had to employ biophysics, which led to the idea of cube homemade microscopy. Touched upon developmental biology by studying life cycle and in turn cannibalism. Lastly, we worked using biochemistry principles for protocol design and media preparation.
Racking up with the further plants.
Research objective: comparing the olfactory sensitivity of standard CSBZ and native Drosophila melanogaster larvae. So what's next? Standardizing the TRSV medium, improving the cube homemade microscopy, moving further with identification of native single lines, making an alternative for plain agar plate, using Rava for developing the cube homemade olfactometer to perform olfactory assay.
Yeah, so we can start with the discussion. We can take question. Any question from YouTube and chat?
Yeah, there's a question asked like, what is attracting the fruit flies? What is there in the fruit?
Hello. So as you all see that there were so many different fruits to which fruit flies are attracted. There was tomato, mango, but then what is the commonality between all those things? Is that the fruits when we keep them open, there are chances that the fruit gets rotten. And maybe also the fruit has some kind of a smell, that smell which attracts the fruit flies. Usually it is alcohol that attracts alcohol, or acetic acid, as they say, is one of the chemicals that attracts fruit flies. So it's not the sugar or not the color of the fruit maybe, but the smell that it gives. Like the sweet banana smell that you get is attracting fruit flies to banana, something like that.
Okay, so there is another question. Why are we comparing the native fruit flies to the standard CSBZ flies?
Because we are comparing with Drosophila melanogaster from which are in lab, and other we are collected from our locality, Drosophila melanogaster. So first we want to identify our locality fruit flies, which of them are Drosophila melanogaster, and then we want to identify, because there is an hypothesis, while in our long-term objective is to identify olfactory test using second instar larvae of native Drosophila melanogaster and as well as CSB fruit flies, like CSB set. Yes.
Yeah,
Thank you.
actually, my question was why we want to compare standard and CSBZ via olfactory.
Yes,
What is
that's
the
why I'm trying to say, because that's because CSB said fruit flies they are culturing 70 years old. I guess yes. I'm not wrong. Yes, and while we trapping fruit flies is Drosophila melanogaster in native fruit flies, it's because our hypothesis is that one it's not possible. Yes, that's one is CSB said fruit flies have a less olfactory sensitivity comparatively native fruit flies. That's why we are
why you
just
think so
trying.
that why you think so like CSB jet will have a less olfactory sensitivity as compared to the native thing?
Because why? Because yes, that's why I'm just earlier tell that 70 years old tradition in CSBZ fruit flies. So and they are also they have a availability of food. This is just a hypothesis and we want to prove it, and they have a they have a food, standard cornmeal agar media we are given to them, but in the case of native fruit, native Drosophila melanogaster find their own food. So comparatively these two, just a hypothesis that these two flies have a CSBZ fruit flies have a less olfaction because they have a availability of food. Yes, yes.
Yeah, I also wanted to add Ashwati's point that when she was mentioning CSBZ, that is one of the Drosophila melanogaster species that is cultured since so many years. But what the catch is that for so many years it's in the bottle, meaning all of its generations. So if you trace back more than 3,000 or 4,000 generations of those flies, are cultured in these bottles. And what happens is the hypothesis for the CSBZ flies having less olfactory smelling capacity, as we say, is that there could be a chance that because while there could be a chance of mutation in one of the genes that forms the receptor for the sense of smell of the fruit. For example, if there are starting if there are 10 flies, out of which even if one fly gets a mutation can happen as a random chance that one mutation that causes the change in the structure of the receptor. If someone can keep the whiteboard or the presentation on, or I'll just try to draw that thing here in the space.
Yes, I have one thing to add. When you talk about mutation is a random process, as you're saying that mutation, there's a possibility of mutation in the CSBJ flies. So there are equal number of possibilities of having a mutation in the native flies as well.
Exactly, you're absolutely right. Absolutely right. So you're saying that CSBZ flies, there are ones, and then there is the native that we are trying to trap. In CSBZ, what is happening is all flies that suppose one fly got this mutation in the receptor of that particular that smell molecule. If suppose this is banana, I am trying to draw a banana molecule here, and the receptor that is coded by some gene that is coding this is the gene. If there is a mutation that changes the structure of the receptor in such a way that it is not now able to bind to the banana molecule, okay, and what will happen is it will not be able to sense the banana. This is not happening. Okay, so this is happening if suppose this can happen in the native flies as well. But the catch is that in the bottle culture, all those mutations are passed on to the next generation. So meaning in the sense all the food is already available here. Even if there is a mutation that causes it to not smell the food, still the fly will survive. Hello, am I audible?
You lost your voice. Yeah.
Yeah, sorry. I'll just repeat. I was saying that even if there is a mutation, as Vivas was saying, that there could be a mutation in the native flies as well. But if there is a mutation in the CSBZ fly, that mutation will be passed on to its next generation without harming in the sense, if there is a mutation in the native, in the wild.
Yes, in between your voice is breaking.
Yeah, is it audible?
Yeah. Yes. Meanwhile, someone else can add maybe. If you can try rejoining. So yes, Bivas, you were saying that there would be equal amount of chances to get mutation as it is random, right? So yes, that's so true. But then in the CSBZ culture, there would be no natural selection. As in, what do I mean by it? If there is a mutation in the olfactory sense of the fruit fly, the food is already available. They don't have to use this sense of smell to go towards the food. So there would be no selection there. All will survive. As in, fruit fly having less sensitivity, fruit fly having more sensitivity, fruit fly with normal sensitivity, if I say. And in native flies, supposing there is a mutation in the fly where the flies are more sensitive to the smell, it may get selected over others, as in if it can smell from like much like from more distance then
Saida, suppose a situation is like if there is a mutation in the Drosophila which is living in the native thing, and it has changed the receptor
Devas,
for
I can't
olfact.
hear you in between.
Can anyone can you hear me,
You
Saida?
yes, I can hear you clearly.
Yes,
Okay.
Devas.
Yeah, so if there's a mutation in the native Drosophila, and there's a change in the receptor as Yash was speaking, and suppose that
Can you
Drosophila,
write it in the chat box? I can't hear you. In between, I'm losing your voice.
everyone can hear you, Saida. I think there's a problem on your side, I guess.
Okay, I'll write it.
Yeah, so what I was saying is like if there's a mutation in the native fly and there's a change in the as he has said that there is a change in the receptor that is getting that particular odor particle. So but if that particular native Drosophila is living near the food source, then do you think that the natural selection forces will work?
But it is very unlikely that food will be so easily available in nature.
Exactly, Jai Kishan sir, because what we were trying to just address as an hypothesis is that the CSBC's Z fruit flies are already having an environment, they are thriving in an environment where the food is already provided. But in nature, food is everywhere. Like the native fruit flies will have a better olfaction than the CSBZ.
No, but is it because of food is everywhere? Where is food everywhere in nature?
I meant like
for the food.
Yeah, that's right. Sorry.
There is so much
They want to search
unpredictability.
their food.
You are daily feeding them with the food.
Yes,
Yes.
exactly. There are like
Yes.
in the hypothesis itself, there are sort of assumptions we are taking. Like both the possibility is there. No? I want to convey that only. Like there is a
Yes,
possibility of
Devas.
both.
Definitely, it's a hypothesis. We want to prove it, but maybe it's definitely wrong.
Yeah. So it depends upon whether place to place. There can be some place where there is no food available, right?
Yes, yes.
But I don't know whether is there any place where every time there is food available. Like they will have to find out food. Like they will have to find out different places every time. They will have to roam from here and there. Then only they will be able to search the food. It will not be like provided to them in nature anywhere. Like you are providing in the bottle. Today it can be somewhere, tomorrow it can be somewhere else, that way.
So the basic point of this hypothesis, the reasoning behind, of course, this hypothesis is not, it's just a hypothesis, not yet proven. But the reasoning behind this hypothesis is very simple, that in CSBZ, there would be no pressure, there would be no selection pressure, because the fly don't have to smell and go towards the food is readily available. If there is mutation for the where this
But
flies
I think if you talk about general pressure,
Yes, Devasi.
what I think is like if you talk about any pressure that you talked about, a native fly, like bringing a native fly to, like as we were also questioning regarding the domestication will phase the fly into the more stress condition.
So how is that related? As in, can you
Yeah, you said
Different
that
for
there
the
will be
person.
less
Yeah.
pressure. I don't know what pressure you're
Oh,
talking
so,
about,
so
but you said that
wait. No, not pressure as in that. Natural pressure as in selection pressure as in, okay. So if
As
the
in to
food is
survive.
readily available, the fly don't have to smell and go towards the food. Okay. If there is a problem in the sense of smell, as in I'm just saying problem as in if there is a fly which can sense the smell from a distance, that can also survive in this environment, and the fly that cannot sense the smell from a distance, that can also survive in this CSBZ culture. Whereas a native, they have to smell and go towards the food, right? So uh, so if
So it is
there is
similar
no,
to what Vivas is also saying, right? In his comparison between
yes,
native and lab bred,
yes,
he also is doing the same thing, right? With
yes.
yes,
So I was just trying to bring that point only. Like, is it the same thing that we are also targeting for the C. elegans as well? And what if, like, if you prove that there is a change in the olfaction between CSBZ and a native fly, then what next? What are you concluding from that?
So is it a repetition? I'm asking another question. Is it a repetition of C. elegans, or is it something different from C.
Yes, sir.
elegans?
Yes, sir. Definitely, it's a repetition. There is no difference
Why
between
repetition?
C. elegans.
Why repetition? Why
Because
no?
yes. Okay, sir. We are okay. Our experiment is doing with second instar larvae of fruit flies. So approximately second instar larvae is 1 mm or 1.5 mm maximum. So in the case of C. elegans, also same. Okay. What I am trying to
not
say,
about
both of
the
them are
size
warm.
totally. Yes, it's about
And
the
why
process.
particularly
As
second
in,
instar larva? Why not adult Drosophila?
because
Because
yes,
because
Ashwati.
they okay, thank you, Saida. Because they are most hungry larvae according to us. We are okay, but we understand that because in the case of 1 mm larvae, it's very minute, and I mean 1 mm. Sorry, first instar larvae is very minute. We can say 0.8 mm to 1 mm maximum, and in third instar larvae, it's also growing well at approximately 3 mm, and they are very lazy because they are started to go into pupation. So we want middle of larvae, like that's why we chosen second instar larvae. They are most foodie and not much developed like third instar larvae, and
But
so that's why.
does like when you tell about like they are more foodie, does like in the second instar larvae the entire olfactory system is being developed? Like, I'm not aware of it. Is it developed in like fruit fly in the second instar larvae, or is it better to use a adult larvae where the entire olfactory system is being developed?
Okay, Bevas, why we are selecting second instar larvae? Because they are most hungry larvae, and while we put into any bird, they are searching their food ourselves. If we are put into in our olfactory measurement, we are put into them in a middle. So we put anywhere our essence of some banana. They are searching their food themselves. They are very hungry. So I think in the
Taking
case
up
of
your
three of them,
point, so
Your
I
answer,
think
like we couldn't get it.
the answer would be that the olfactory. I think the olfactory sense is developed in second instar larvae. We should definitely
yes.
find references to it. And why we're not using fly comparatively is because if we use fly in the olfactometer that we have designed, it can fly over. We will not be able to see where the fly is actually.
Saida, one point to intervene, like why we are only focusing on like olfactometer, why we have to traditionally use that olfactometer? Because if if the olfaction is not very well developed in the second instar larvae, and if we take Drosophila as a model organism to try for, and where the olfaction is
yes.
well developed, then we can make our own olfactometer. Why
Yeah,
do we have to make
Yes,
it?
Vivas, we are also
Right,
trying
right.
that way. Yes, we are also trying it in
So
our
yeah,
home lab.
actually, yeah, Ashwati can talk. She has also tried to develop olfactometer for flies, fruit flies, and in the lockdown. But we have to. It's like a further plan. But to answer Bivas's question is that is the olfactory system developed in larvae? So yeah, I think so. There are a lot of references. I will quote a few. One is, I think so, Ashburner's reference that talks about how instead of using a fruit fly, we can use a larvae because its olfactory system is simple. So they quote it as to be simple and it's well developed. We can mention it through one experiments that we see do in the labs, that the olfactory assay, where if you keep the larvae, I'll try to annotate. If you this is kind of the olfactometer. If you keep some larvae in the center and you cover this meter, it's like an agar plate, and you keep larvae in the center, and on one side if you put its the smelling the banana smell or banana juice, and one side it's water. Most of the larvae, if 10 of the larvae are kept, most of them will be going towards the banana. So that this is one experiment that can say that it is well developed somewhat.
This is using in lab, I guess.
And also one more question here.
Yes.
Yeah. Also one more question. Yes. If as you said that if you're putting like banana juice at one end and water on the one end, and if the maximum number of flies are going towards the banana juice, if I get a condition in which none of the flies are moving, then what will be your conclusion from that?
That would be the end point, right? See, when we talk about smell or hearing also, so there would be some cutoff point. You cannot smell each and every molecule.
Okay. So I understood about the cutoff point. Suppose if the olfaction, the fly you are talking about has a good sense of olfaction and it started moving towards the banana juice, but it can't move because it has some mutation in the moving system or the muscle system, then how are you going to conclude that whether the particular fly that you have is like having less olfaction?
Okay, but one
Yeah,
fly
that's a good question. So maybe we'll have to because this is another mutation that you mentioned. Maybe we'll have to do some another assay to minus or remove that aspect of whether the fly has a mutation or its movement that causes this disbalance in the movement. There could be some other assay that like climbing assay that can be used to confirm.
Yeah, because why I asked this question, there's a question in the chat where Jay asked like why not check other parameters and why only olfaction? We have dilemma.
So in terms of like nematodes also we are doing olfaction. I think that's why
it's a repetition. So what I'm trying to say, see, if you are repeating, see, there is no problem if you repeat, but you have to make something sense out of repetition also. So for example, if you are repeating the same experiment in another model system, so why you are repeating? So do you have any problem if somebody studies olfaction compares olfaction in nematode? No. So you can say that either you are comparing olfaction between nematode and Drosophila. What are the things what are the differences or similarities between olfactory system in Drosophila and nematode? One thing. Or you can say that you can think little broadly. You can check other parameters also. Like you can compare other parameters like between native and lab bred. Can there be some other differences also, like not only olfaction? Can there be like
we are taking
let me complete, Saida, 1 minute. So for example, do you think that this domesticated one Drosophila will be having a mild behavior or kind of less aggressive or something like that? Their behavior will be different as compared to the one which are having outside, so they will have to fight a lot. They will be kind of not very much they will be aggressive or like that. But here they are a little bit friendly like that. So there will be some social behaviors also which you can study between the two. So you don't have to do anything. You just have to do different type of assay, and you have to check different type of parameters, and you can make some story out of it. You can make research questions out of it. That.
And adding to Jay,
you will be open. You will be thinking openly, more open. Yeah.
Yeah, adding to Jai, what Yash also mentioned, that can there be behavioral differences? I think there will be, because considering you are culturing so many flies in a same single bottle, and whether the native flies doesn't like compare that much of competition, so there will be like a lot of behavioral differences. And so what Jai was also suggesting, because we came across the same type of question when we were thinking of making the olfaction in nematodes. So one last question to the team is like, okay, you found out that there is a difference between the olfaction between CSBZ and native. Then what after that? What do you conclude from that?
No, like same thing. Yes, you can. Deepika.
Yes, so what we could understand, what we will understand from that analysis would be that the CS-based strains would have a less olfaction and the native would have a better olfaction. With the results, we would conclude, because we are the thing is that what is our understanding is that in nature, the native fruit flies, there would be natural selection, and people have talked about it. But in the bottle, it's only the progeny which has from which that has been started, that is was 70 years. It could be the same lines. There would be variants, but it would be the same lines. So we expect, as Jai sir had suggested too, that there could be some behavior changes too. So including that, we would want to also study the olfaction bit.
So you can make us say that also, that the behavior of olfaction also is actually decided by the environment. Like based upon the environment, the behavior is influenced by it. So you can check. You can say that it's not that just that because it's not in the fly. Like they are both are the same flies. They are both the same species, but they are because they are in different environmental conditions. So that environmental condition affect the organism, and they decide the being of the organism. They decide what actually the organism will be, what kind of organism it will be. So that being thing also can be decided by the environment. So you are bringing into lot of things, like you are twisting the whole thing, and you are bringing the context of environment and how environment affects the behavior.
And also adding to Jai sir, and also what we faced in C. elegans when we are planning to like nematodes, when we are planning to do the olfactory assays, like will it be possible by only doing an olfactory assay to conclude this conclusion that Deepika was talking about, or will we have to consider many other factors as well? So this was one of the major questions that I came across when I was thinking of doing it on C. elegans.
So one of the major question that nematode group is asking, that is the C. elegans, the standard strain is really standard. We can ask a similar type of question in the CSBZ has been cultured for so many years, but we expect that there would be mutation, there would be changes in the fly. So can it be considered standard?
Yeah, that also we can see that. That is true.
But that cannot be compared based on the one assay. That's my question.
Yeah, that is also good. Yeah, that is also good. Because one assay we cannot say whether it is standard or not. Because
right. So yeah, like today's in the discussion, we got a lot of inputs into how we can develop more assays.
You can do all these things into the home lab. Like your all experiments you can make for in the home lab. You can find alternate. Okay, so I think let's
C. elegans and fruit flies collaborate together.
Yeah, they should collaborate. Both of the group. I think is there any other question? So we can take it on the group, because the time is actually like 10 o'clock. So I think people will have to go for dinner. So last thing is Prashant, you can write in chat box. We will raise it. You have any difficult question or what? Bigger question?
I am saying that. No. So how can we this Drosophila can help us to sense olfactory sensor?
No, we are checking that how the organism actually evolves.
So rest of the questions
I
can
think
be
we
taken
can take
in
it in
the
the group.
group.
You can ask this question into group. The people will be explained. Now only you can write. Just put it in the group. Now only people will be responding to it. Okay. So let's move to the next. I think DP Durga Prasad. Let's move to the next question of the mango. I think we have 15 minutes. We'll finish at 10 o'clock.
Yeah, so I think we will, sorry for delaying for all the participants. So we will quickly try to show certain things and reduce the workshop part. But I think this is going to be a quick trigger of a lot of activity starting for the next 3 days and continuing ahead. So Dristant, if you can quickly take presenter and upload the slides.
The slide on the screen.
Yes.
Yes, Tulika is doing that. Yeah.
Okay, great. Yes.
Are the slides visible?
Yes, they are visible. Thank you, Tulika. Yes, so basically, I think there is a lot of discussion and there is a lot of excitement around eating mangoes, and that happens in summer. But we often forget about a lot of things that happen to mangoes all through the year, and okay, we can continue mango tomorrow too if it is not finished.
But you can continue, DP, let's see.
Okay. So what we have is, okay, let me take presenter and change the slides. Yes, Dristant, feel free to jump in and say things in between. And yeah, so what is mango mapping? So basically what I was saying earlier was we forget what's happening to mango trees and others for the rest of the year. But the interesting thing is there is a lot more that mango trees can offer us other than just mangoes to eat. And that's what we are going to see with the mango mapping. So quickly, like maybe in 4 to 5 minutes, we will finish what we want to show. And then I think we will have an open conversation connecting it with the curriculum. Yes, Dristant. So what is mango mapping? You want to say something here?
Yes. As the mango season has now begun, that from September, all of us have been posting many photos of mango trees from all our localities, and we have just conveyed the same message to our relatives and everyone around us. And the groups are always filled with mango trees, mango tree photos.
Yeah.
And the essential data with that. So we need to understand that why are we actually doing it? Are we doing it for ourselves, or we are involving everyone in that? So and we need to make them realize that what is the importance of studying this mango mapping. I mean, mango tree studies.
Yeah. So from what I understand, I think mango mapping can be a very good initiative to draw lots and lots of students and the young people to ask questions about
citizen
things
scientists.
happen. Yes. They can very easily, as Dristant just said, like all they need to start with is look for the mango trees around them, and they just need a mobile camera and a very observant eye, a very sensitive eye to what's changing in the nature around them. And this is, I think, will also increase the sensitivity towards nature among the students. And there are quite a lot of interesting questions, research questions that can be asked given the diversity that our country has, both in terms of geography, especially in terms of geography, I think, and weather, so from north to south, and there are various landforms and others. So how it's going to be a very interesting research question as well to see how these things change with geographical conditions, and that definitely requires collaboration, not with 5 people, not with 50 people, but with lakhs of people across the country. And hence, this is going to be a very, very great and wonderful way of engaging millions of students across the country. And I think we already have a small trailer kind of a thing through our interactions on WhatsApp groups and others, where everyone, like from young children to old grandmas and others, they are sharing very enthusiastically a lot of photos and others. And we see these photos from different places, from Bangalore in the south to Patna in Bihar. So we see leaves sprouting in, as you can see, it's October 27th. And we don't have a date here, but we will have them. We see flowers in Thrissur, in Kerala, and a fully flowered mango tree in Ranchi, and fruits already in Kerala and in Ranchi. So I think this is the very interesting model system where we can ask multiple research questions. We can engage with multiple people of different backgrounds. And if you just were here a while ago, we looked at weather station. So if we have weather station and we have mango trees and others, and see correlation between what parameters vary the behavior of plants and animals, so we start becoming sensitive to how alive the world around us is. Does anyone want to add anything before we sort of open it up to conversation? We also have a context to curriculum thing where I think Drishtant sort of connected some of the.
If you have maps to show, you can just show if map is there. Some map.
Yes, it's on the next slide. DP.
On the next slide. Yes. Yes, we also have some maps. Sorry for that. Yeah. So there are. So this sort of shows very quickly where the flowering trees, where the fruiting trees. So you want to add something, Jay, here? Jay or Drishtant, anyone? Yeah.
Yes.
No, I think you continue. Yeah, it's showing very evident.
Yes.
Yeah.
Map.
So this map can. Yes. Can I have the pointer, DP? Yeah.
Yes.
Okay. All right. Okay. So this map shows the diversity of what the mango trees have. Now at the south, we see that there are some yellow marked and there is some orange colored also. So these yellow colored marked trees, I mean the yellow colored icons, it is for the flowering of the trees. Mm-hmm. That means the flowering has started. And in between, we see that there is a green mark that is in Palakkad, that is in Kerala. So in between these flowering trees, one of the tree has started fruiting also.
Okay.
That means mango fruits have been developed from there.
Okay.
The flowers. And we go a bit north in Goa, the trees have started sprouting new leaves there. And then if we go to Visakhapatnam in Andhra Pradesh, we see that the mango fruits have been fully developed there. I mean, they are hanging behind, below the trees. And as we move northwards, we see in Rajasthan, there is no sprouting at all seen till now.
Okay.
Like the sprouting percentage is very less.
Okay.
And if we see in the northeast side, say in Bihar and Assam in Silchar and in here Jharkhand, we see that the sprouting of new leaves has just started. So an interesting observation can be made that though Delhi, so this one which I'm marking, it is Delhi and here is Moradabad. So we see that, and here is, I think it is from Madhya Pradesh. So we see that Moradabad and Madhya Pradesh fall on the same longitude, but there is different scene in that both the mango trees. And here New Delhi is close to Jaipur here, but no sprouting of leaves is seen. So why is this change happening? Is it because Rajasthan is a kind of very arid area? I mean, desert? Is it like that? Or what is the actual phenomena happening behind both these observations? Though we are observing the mango trees. Like both the trees are same, but two different phenomena are observed here.
Yeah. So how many data points that we have here, Drishtant, like in this map that we are seeing right now?
Yeah,
Roughly.
so we have, yeah. Okay, so we have the number of trees surveyed, first one. And the second one is the number of trees fruiting, not fruiting, sorry, sprouting leaves first. And then the third one is flowering.
Okay.
Fourth one is fruiting. So fruiting is a later part, but yeah, we should include that.
Okay, so I wanted to know like how many observations, like how many individual tree observations are mapped right now in this? Is it 50, 100, 1000?
No, I think it's 72.
Okay, so it's with just 72 observations, like this is the kind of insight that we can have. And if we have all of us, all students, all people, like when we go out, like we see mango trees all around us. And if we can engage it as a project or as an activity at mass, it's something called citizen science kind of activity. Not just passively collecting data, but engaging in a discussion around why we are collecting the data and what questions we can ask with it. It's going to be a very interesting and live context for us to discuss quite a lot of things as against the boring school and college sessions that all of us are subjected to.
So far 490 trees have been mapped.
Yes, 490 trees. Yeah.
Yeah.
I think if any of the participants have any questions, we can take questions for 5 minutes, and then I think we will quickly show a demo using which. Yes.
Yeah, we can move to the context to curriculum map so that questions will
Demo
arise.
you want to show, you can show it because people are there, still 41 are there.
Okay. So maybe we'll show the demo first, and then we can continue the conversation. Is that okay?
All right.
Yeah. So Drishtant, Jay, and I, we discussed yesterday, and we right now try to sort of get a picture of how this data is being collected and how it's being managed. So right now we have lots and lots of people sharing things on WhatsApp. And as you can see, the name, place, and the time are very important parameters or very important things. It's called as metadata, that if you want to say, for any observation, whether it's flowering, fruiting, or whatever. So we have been, Drishtant has been manually putting those things into a spreadsheet, and it is a very tedious thing. So what we thought is, especially the ones who are pretty regular among us and who have been doing this for quite some time, it's time now for us to become a little more rigorous in our activity instead of just taking a picture. So we can take a little more effort so that these things will become collated in a single place. We can still encourage people to send pictures on WhatsApp and others, but slowly we will nudge them towards pulling them all together into a particular space. And to do that, like we developed a simple form, the link to which again we will share, and Drishtant has used it for the last 1 or 2 days, and his experience was smooth. And it has very simple set of questions. So name of the reporter, you will say whether the mango that you have observed was flowering, fruiting, or sprouting. And if others, you can select others and write the observation. You can mention the location of the place, and you can, instead of manually typing in the latitude and longitude, you could, if you are using your laptop or machine, you
can show the form. You can show the form, our Mango form.
Oh, sorry, I didn't share the screen. Yeah, thanks, Jay. Okay, sorry for that confusion, and this is the link that I just shared in the public chat. So you can open it on your phone or on your laptop without any issue. You will get this page, so you will write the name of the reporter, whoever is reporting the observation, the type of observation, so whether it's flowering of mango, fruiting of mango, sprouting, or if any other, you will select other and write it, mention it here. Location of observation, so you can write whichever place, so say it's Ranchi, you can write it here. And if you can leave it right here, but if you are okay, you can do the same thing here as well. You can just type in and search, so it will show the map of Ranchi, and you can just map, put a point anywhere, and the latitude, longitude automatically comes here. Or else, if you are using your mobile phone and if you can enable your location, you can just click on this, and it will automatically select where you are. So that can also be done. And the date and time, so you can on your mobile phone, like normally the way you set your alarm, like that interface appears, and you can quickly select the time and date, and then you can just tap here, and if you're on your mobile, your camera will get opened, or you can upload image from your gallery. And because I am on my machine now, so it's opening up a browser browser on my machine. And then you can save it if you are in a hurry, or if there is some data issue at that moment. The data, if you just click save, it will locally get saved, and you can open it again and submit. If you have no data issues, you can directly submit. Because why this option is there is, most of the times we don't see mango trees inside our home or in concrete jungles. So we find them when we are traveling outside, and some places signals and data issues might be there. So even without bothering to worry about it, like you can just click and save. So it will be saved here, and you can submit it once the data is back. So I will now quickly demonstrate one or two points of one or two.
Just show one example so that it will be clear.
So Drishtant has shared with me some photos. So say this is the photo that I have. I will download this. So this got downloaded, and I have the data collector's name, which is Akash Panay. So Akash, is that correct? Akash Panay. And yes. It's Seawoods, Navi Mumbai. So let me just, before Seawoods, Navi Mumbai, what is the type of observation? Sprouting of leaves, no flowering. So it's leaf sprouting. Yes, leaf sprouting is selected. The location is Navi Mumbai. You can just leave it as it is if you don't want to give exact latitude and longitude. Others are not made mandatory, but you'll have to give a space so that rough location can be made. And you can do the same thing here. So let me check if Seawoods is there. Seawoods, Navi Mumbai. I guess it should show up. Okay, so it has showed up. Seawoods, Navi Mumbai. You can, if you are very particular about exact space, you can zoom in and zoom out and select wherever you want, and you just need to tap. So the latitude and longitude now has changed. You could do the same thing using this icon, and it automatically asks for your location and it gets populated here. And the date and time of observation. So it's 2nd November, and I'm assuming it's daytime. So 2nd November, and I will give some AM time. And point and shoot, use the camera to take a photo, or right now I'm going to upload the picture. And I can save the draft if I don't have internet connectivity right away, or I can just click submit. Yeah, so queued records except those marked as draft are uploaded automatically in the background every 5 minutes when the web page is open and internet connection is available. So it's just giving a message that the image is saved whenever you have saved the draft locally, and the moment you have internet is there, it gets submitted. And you just saw a symbol here saying that it got submitted. So yeah. So the initial thing got submitted, and when I try to submit it again, it's saying that it's a new observation. So and so this is how it is. So when you use it 1 or 2 times, it's very simple. After that, it's going to be very easy. Your name, select the option here, write the location here, select the time, and pick the photo. And if you have more time, you can select the space here, and the latitude and longitude will automatically get captured. Okay, so this is how the form is. Now, how would the data look like? So let me reload to see if there is some new data also that got uploaded here. So the data, because it's going to be rich data, different kinds of formats are going to be there. Yes, we have one from Akash Panay that got added. And there is a lot of data that Drishtant has already submitted. So you can quickly see some basic analytics like how
this
many
data is open data, like everybody can view this data.
Yes. I can. I'll have to just check if it is made open yet, but we will make it open. It's going to be visible. So this is quick report of where all things are happening. You can see it as a table as well. So you can see, you can download the image from here. You can see the location markers. You can see the time, date. You can see the location here, and you can see the type of observation. So it's like a Excel thing here. So this is just to view, and you can see it as a gallery. So all the images will be visible here as a gallery. So at this stage, in this format, you don't see the metadata, but in other formats you can. And you can download this data in multiple formats. So Excel is the normal Excel format, but because this data is pretty rich, there is a lot of metadata. So you can download it in any other forms for later analysis. And then you can have the map feature as well. Right now, it just shows number of observations in different places, but depending upon what kind of observations each of them are, like we can modify this particular view, or we can export this data and represent them just the way Drishtant has showed in the presentation available. So those things can be taken care of if all the data is sort of pooled at one place instead of spread out in different places in WhatsApp groups. So
also we can they can share on WhatsApp images also if they want, and they can
yes.
It's both the
Yes.
things they can do.
Yes. So WhatsApp images. So that's what we were saying. All the students and all the participants who have been doing this regularly, we can ask them to directly submit it here. And those who are newly joining, we will ask them to, we will let them share the images on WhatsApp itself. and we will create a group team of people from among us who will regularly keep using the updating the same images, just the way I did. For example, I'll take another picture. Suppose this is the message that I have in WhatsApp, so I will download it. I will take the metadata from here and I'll just fill in the form, whoever is doing it. So that first, we need a set of people from among ourselves who can frequently, like once in a week or twice in a week, sit together and put these things in one place. And yes.
Yeah, DP. I think so we can also use STEM game. They can create some threads there also. So
yes,
if they have a large number of data, where they're putting on their WhatsApp, they can same thing they can shift there also. Like if possible, somebody can put it on. First, they can put it on STEM game, then they can share on the WhatsApp. That is also possible. So
yeah. So let this be shared here. Baki WhatsApp pay or STEM games pay, like that's completely up to them. But this is something that all of us need to make sure that data is getting collected or data is getting uploaded here. And यहाँ से जो भी दिख रहे हैं या updates from here can continuously be fed back to WhatsApp groups or STEM game place, so that people know. People can see that what they are contributing is actually being useful towards a particular research, and they will also that becomes a good context for them to ask us questions. Okay, what is happening to the photo that I sent? What does it mean when I say that when I observe that it's flowering? And they can see, okay, so just the way I'm doing something in say Bangalore or Kerala, and there is someone else doing the same thing in Delhi or Ranchi or say Northeast or anywhere, and they can feel connected with a larger research activity that's happening across the country. Yeah, so that's what we have, and the idea was, if we had time, we wanted to do a hands-on workshop or hands-on activity where all the participants who are currently here will try it on their own machines or on their own phones. You could do it after this session, and tomorrow again, like we can come back and maybe we'll have a quick short video showing the demonstration of this, and troubleshoot if any of you are having any issues. That can also be done. Yeah, so is there anything that any of you wants to add, Arunan or GN?
Any questions?
Yes, any questions?
No.
Yes, Prashant. Yes, all of you are hungry, and some of you might already be sleepy because you have been having sleepless nights for last, I should say, maybe a week or 10 days. So, don't worry, we will come back to this thing repeatedly, like at some point again tomorrow and day after tomorrow. Keep sharing this in your groups, and Drishtant, please gather a group of people as volunteers. So whoever is going to volunteer, please contact Drishtant. He will be the contact point even after this meet, so that we will continuously keep working or keep meeting once in a week or twice in a week to sit together and collect all the data into this. So right now we just have it for Mango mapping. We will see how smoothly this happens. But once this is done, or once this sort of people get used to it, we can expand it for other observations as well, including Phyllanthus, sleep wake cycle, crow nesting, coil cooing, various other things. Yes, we will repeat this again tomorrow. Yes, so I think that's it then from the Mango mapping workshop, if there are no questions. So over to you, Jay.
Yeah, so if there's no question or somebody has any question, then they can also write in the chat, our chat, or on the group. Hello? Hello, DP? Durga Prasad?
Yes, Jay. Sorry, I was on
So
it.
yeah, so I'm saying that so we can have more question. If somebody has, they can write in the group, and
Yeah,
we can
yeah, yeah.
take on the discussion.
Sure,
Because
sure.
this mango mapping, we can also show the form tomorrow once again,
Sure.
and let other people also who are joining tomorrow, let them
Yes.
also get to involve this. So it should not take like 4 or 5 minutes, more than 5 minutes.
Yes.
Take a break
So
time
very
or
quickly.
gap or something, some gap. Okay.
Yeah, before the close of the meet, I will try ensure that it's shown at least half a dozen times, so that people are.
Okay. So I think Arunan sir is. I think Arunan sir is back in the meeting. Okay. So I think we should. It's already late. I think we should close the session for today here.
Yeah.
Is there anybody has any question they can raise, or they want to say anything for today's session? We can have some views up from the participants, and
Yes.
then I will like to call Arunan sir for just giving like few words
Concluding.
before ending the session, today's session. Anyone has to say anything about today's part from participants? How was from today's session? Anything you can share, your views, any feedback you want to give, or anything you want to say about what did you like or dislike, and like.
Also, Jay, should we stop the stream now, or we will continue?
Let's have for like 5 minutes. Let's have the
Okay. Sure.
Then we can.
Hello.
Yeah.
Hello?
Say. Yeah.
I think that we have to create some feedback form.
In a short, you have to say in very short because it's already late.
Yeah, he was saying we'll have to create feedback forms. Okay, Prashant, we will look into it.
So for now, we have seen how people have developed home labs, making media, studying about oscilloscope, how to make oscilloscope using phone. So I think if I was to summarize the session, we could say it was collaboration, understanding, breaking down things and understanding and creating it at home. And yeah, we would continue to do so, and in coming up sessions in rest of the 2 days we'll look up more about what we can do at home. So this is not just for us, as we say that we collaborate, people who have joined through YouTube, people who have joined through Chatshala as well, can join our group and work together, because that's what CUBE stands for. It's not me particularly working on something or a group particularly working on something. It's a collaborative effort. So yeah.
Also, I'm looking forward to the new venture that we actually found out, like collaboration between Nematode Group and Drosophila. I will be really looking forward, and we all will be looking forward to that. And I think I really connected with the study of Drosophila, the work they presented, because it is much related to the nematode study. So hopefully we can continue this collaboration.
Yeah, at this juncture, I just also want to highlight that we also need to start actively thinking of working together with makerspace groups and people beyond biology also. Yeah, we will also try to find ways of nurturing such collaborations. So we already saw Snail Group has reached out voluntarily and they tried to do something, and so we will be having a microscopy workshop as well tomorrow, very briefly, which is going to be just an initiative or initial trial kind of a thing for more or a series of sessions around microscopy from next week onwards. Yeah.
Anyone else also can share? Anyone has to say a little bit, anything, feedback or something?
If no, then we can wrap up, Jason.
Okay.
I think everybody is tired enough.
I think okay. So I think if Arunan sir is there, can Arunan sir share a few views about the session, today's session, and before we close the session, anything you want to say, Arunan sir?
I think earlier he mentioned that he has some issue with his mic.
Okay. Nagarjuna sir is there. GN sir is there.
Yeah, GN is here.
Yeah, sir. GN sir, you want to share any views before ending? We close the session today.
Yes, I do want to say good night. Come tomorrow exactly at 5:30. Tomorrow we'll make sure that no technical problems will happen.
Yes.
Our setup is now very stable. We are also learning from the mistakes, goof-ups that we do. And so good night, have a nice rest, and then come back tomorrow with more energy. And also invite more of your friends.
Yes.
And of course, we can have more YouTube viewers, though our platform is limited for 100 people. But do invite more people.
Yeah.
All right.
And I just want to mention one more thing, like because some of you may not be aware, or so we also have another set of Charshalas happening every Saturday in the afternoon. So that details are also there on Meta Studio. It's going to happen again tomorrow. So we will. I just wanted to highlight this as during the common session. Yeah. Good night, and thank you all, and see you all tomorrow.
Yeah. Good night, everyone. Bye.
Okay. Good night, everyone. See you tomorrow, everyone. Bye. You can close the session. Who has the control?